Love reptiles? Don't miss this. If you're passionate about reptiles, amphibians, and everything cold-blooded, Reptiles magazine is a must-have. Each issue is packed with expert care tips, stunning photography, and the latest news in the reptile world. Whether you're a seasoned herpetologist or just starting your reptile journey, this magazine has something for you. Subscribe today and stay informed on the best ways to care for your scaly friends. For only $14.99 a year, you can step outside your algorithm and get exposure to things that you wouldn't normally see or learn about. My family reads Reptiles magazine, and you should too. Visit subscribe.reptilesmagazine.com to get started. This is the Expert and the Idiot podcast. If you like the show and you'd like to support us, there's a link to our Patreon in the description below. Some benefits include getting access to new episodes up to a month in advance. There's also links to USARK and USARK Florida. If you're not already a member, please sign up. Enjoy the show. Welcome to episode 61 of The Expert and the Idiot. Today I'm talking to Dr. Steven Tillis of RepTillis Herps LLC. Today we're going to be talking about things like Nidovirus. But first, I kind of like to get some background on you and how you actually got into reptiles and keeping reptiles. I have a ton of questions here for you, but I do kind of want to get to know you more on a personal level of what has kind of driven you to the point you're at right now. I was speaking with Jeremy Turgeon, and he told me that you were like the OG co-host of the Reptile Talk podcast. Like
and that was even
way back.
that was even like 10 years into my herpetological career at that point. Like I've been doing this a very long time. Yeah. So, lived in Florida my whole life. Grew up in Orlando, lots of access to nature, and it's Florida. So there's like snakes through the yard and stuff like that. So the interest definitely started young. Like I have a family of naturalists. And so, it's kind of built into the cake on that. Started with, a pet corn snake, a pet ball python, just like anyone else in middle school. 2009 was freshman year of high school and the first year that I bred ball pythons. At that same time, that was when Barczak was really just putting out some of his YouTube videos and, Snake Bites TV stuff. And along with that came a whole like cohort of especially young kids like myself who are literally just filming their reptile collections at home. And that little bubble of attention kind of grew and progressed as my collection grew. I kind of stopped posting a lot of that YouTube stuff when I was starting undergrad, but still post the occasional video of some of the things that I get to experience on that original channel. But undergrad, I went to college for wildlife, but at the same time was growing this reptile business on the side. That was when I started working for Eugene Bissett. So I managed all of his ball pythons for a very long time and still manage kind of tangentially that colony through the databases and stuff that I built.
Can you tell me how you got to there? I mean, how did you end up working for Eugene Bissett?
Yeah, so that's actually a funny story. So I was vending a reptile show when I was like 17. And I had a green tree pythons that I had produced. I was super proud of it because, it's hard to bring green tree pythons, and to do it as like a 17-year-old kid, I was just, thought I was the shit. So I had them on the table. Eugene walks up, and at the time, so the very first reptile podcast, as far as I'm aware, was Reptile Radio. And that is such a shame that it seems like that is lost media at this point. I've gone looking for so many of those episodes and they're just gone. But like, it's such a shame because there was hours and hours and hours of central people in the industry, kind of towards the inception of it, at least as far as an internet presence. Like, it's been very fascinating to watch the whole explosion of social media and the reptile kind of thing go within that. So the very first reptile radio podcast, one of the most, interesting episodes I always thought was Eugene. And he has a facility up here in Gainesville. And
why did you what did you find interesting about it?
It's really tough to say. Like, a lot of it has to do with the perspective, I guess. You can kind of you'll hear it a little bit when he talks. Like, I don't know. I guess one of the central things to me was always, be a student of the serpent, like really pay attention to the animals and stuff like that. But, even within that, it's more even just being excited about the industry, I guess. Like, I think Gene's passion definitely lies in the industry, and so, his facility is a big facility. It produces pet store level ball pythons at a production quality for pet stores. So it really is reptile industry, and growing that industry and looking at animals through an agricultural lens. And as someone who is, interested in wildlife management, taking, hybridizing that approach of captive conservation, like we always say in the industry, and true conservation, and kind of meshing what that looks like on, a commercial scale, was really sort of the right recipe at the right time of like me. So I'll jump backwards. So when I was 17, he saw these green tree pythons on my table. He invited me up to his facility. I came up to the facility, ended up purchasing a property when I started an undergrad that's very, very close to his, like less than probably a mile away. And so I basically called him up and I was like, I'm going to be moving into the area. Are you going to have any job openings? And it's a big farm. There's always job openings. So I guess not at the time, but then it was much smaller. But so started there really as a grunt worker, working the snakes. Started doing more shows with him, started experimenting with ideas. Like, he really sort of gave me a lot of free reign to manage various aspects of his colony. And part of that was building a really intense database. And it was sort of one of those things, like, we never really had the complete vision of what it was going to be. It was just, how do we manage this a little bit easier? And then it's like, oh, well, once we unlock that, we could do this, and then this, and then this, and then this. And then it turned into like something really crazy. So as I was working in his business, I also was building my own reptile, business at the time, but more focused on, so his business is almost exclusively ball pythons and colubrids. Whereas I was like really into all sorts of pythons. So really built up a diverse colony of pythons. And that's how I built my own, reputation as a independent seller, and sold a lot of blood pythons and, short-tail pythons, carpet pythons, like almost the whole sweep. I really did enjoy, working with all of a diversity of pythonidae on that commercial scale. 2016 is when I graduated undergrad, and ended up just trying to take a go at breeding snakes full time for a few years. But 2018 is when I had the first virus outbreak, at least in a subset of animals in the colony, and really it just didn't make sense, that everything that went into it. And there were so many mysteries at the time, and essentially that's what landed me at the vet school, and discovered what I was experiencing was an outbreak of serpentivirus or Nidovirus. And that sort of got my foot in the door in terms of doing research at the college, and started a Ph.D. working with Nidoviruses and serpentoviruses, really starting from the basis of, so the viral outbreak that I experienced in 2018 affected most of my adult blood pythons, either by illness itself or euthanizing because of exposure concerns, or positive tests, but they were not symptomatic. So euthanized pretty much the entire adult colony of most of the pythons at that point. Had neonates that were in a safe area that were raised up and currently are the backbone of the commercial colony that I have now in one of the buildings. 2020 was when the state changed its laws. And so around that same time, I started getting into a diversity of pythons and really wanted to make a biosecure colony of Pythonidae. Like at this point, so in my viral outbreak in a closed colony, not closed colony, in the original viral outbreak, doing things right, we found like something like 9 different strains of virus. Some of them were causing disease, some of them weren't. And so really we're looking at this giant question mark of like, what the hell happened? And so at the same time, I am worried that many of the pythons we keep in captivity would be vulnerable to these viruses. And so I started building up a diversity of Pythonidae, doing very heavy testing of Nidovirus because I work in a diagnostic lab for my Ph.D. To be able to build a biosecure Nidovirus-free colony of as much of pythons as I could. So I have about half the world's python species at this point in a biosecure building. The state changed its laws that basically says I now have to put a subset of these species on display. And so that sort of created the push behind Archer Reptile Center, which is my educational facility that, is open for the public and the tours for like that you've been on. Yes. And really kind of gave me an opportunity to layer all of this stuff together. So at this facility, I now have different buildings with it's basically herpetoculture through different lenses. So I have a biosecure building, which has almost half the world's python species that went through really extreme quarantines. And I have a bunch of biosecurity themed, how I treat that building is very focused on biosecurity. And that's put on display for people to see what that looks like at a reasonable scale. I have a exhibition building that really has sort of unique ideas I have in terms of managing the animals through housing and stuff like that. And then I have a commercial building that was those neonates that have now grown up, and as well as some of their offspring now at this point. And so I kind of see each of these as different contexts through which managing the animals and their associated microbiomes is treated kind of differently. And so that is the sweep of kind of what I keep now. It is, a fair bit of blood and short-tailed python morphs, so in working with rare species, a lot of them are wild caught. And so it kind of takes some getting used to work with these wild caught animals to get them adjusted to breeding on the opposite side of the world. And so I've kind of extended that through to the commercial species as well, where I'm establishing new color mutations of blood pythons and short-tailed pythons and introducing that into the hobby. And so, all of these, I view herpetoculture as having value on many facets. Like obviously I worked in a commercial facility for a very long time and understand commercial management and disease in a commercial setting. And so that looks and feels and is treated one way. I've worked in a laboratory setting where, extreme biosecurity is very important and very critical. And so how I would treat animals through that lens is entirely different. And each of these has pros and cons and risk profiles and expenses, and, you can really drive yourself crazy with it. And so the goal of this facility is to really put all of this on display and kind of have people figure out that like, this is a complicated landscape to walk through. And the context in which animals are kept kind of matters in terms of how you are approaching disease and how seriously you want to take it and what you want to prioritize. And at the end of the day, you know, I have a whole exhibition building that is just animals on display for me, the enjoyment that I get out of that, and kind of making sure I don't lose sight of what is the why are we doing any of this in the first place, you know?
Yeah. What the one of the coolest things, so you were kind enough to have me and my family out, and I feel fortunate that it was just us, you know, so I kind of had your undivided attention, aside from when my son wanted your undivided attention to ask his own questions. But it was so cool that you're so passionate about them, that you were getting as much enjoyment showing the animals as we were getting from seeing them, you know, which was awesome.
Yeah, absolutely.
You know, you go to some, you know, parks or things like that, and you can tell that the individual is just phoning it in because it's a job, you know, but you could tell your passion and your pride and your excitement to go through the animals and bring them out for us. And it was very cool. It was awesome.
Yeah, I certainly have found my way into a very interesting life, and really appreciate the opportunity of getting to share what is my life's work at this point. You know, it's been 2009 was my first year breeding snakes. So what is that, 16 years now at this point?
Wow.
And the industry has grown a lot in that time point in terms of what it looks like and how people interact with it. And like, yeah, it's been very fascinating. And I consider myself very lucky to have, even though it's sort of a weird set of circumstances that have bounced me into the position that I am, I really do have a unique position to mix all of these experiences and perspectives together. And you know, my goal is, I don't think I've ever had anyone come here without learning at least something, like something they just never knew. Like there's so many weird things here, or normal things put in a weird way, that I'd like to think that it, you know, it passes along that excitement that gives people permission to understand that the first spreadsheet, you know, the first care sheet you read is maybe not the only way to do things. Like there's a lot here.
Yeah. I think I definitely learned a lot coming out and seeing you, but I think I left there with more questions than I came in with. You know, it was there was a lot more. I think I finally understood when we left there how nuanced serpentovirus is, and you know, you had kind of mentioned that you had animals that were not clinical, they weren't diseased, but they were positive back in 2018, right? And those animals were culled. How would you approach that now? You know, having animals that are positive but not clinical.
Yeah, so it really is. It really just matters the context that animal is in. Like, I really want a tight control of what is coming into the colony and making sure that is negative. And so in that regard, I would not accept a positive snake with an unknown virus as a risk that I would be excited to take on. Whether it's asymptomatic or not, because we really just don't know what the introduction of novel virus would do to a colony. In the context of an existing colony that has a track record of very healthy animals without active disease, I view a positive test as much more likely to be kind of a harmless part of the microbiome, which is what we've seen in Burmese pythons in South Florida, where we see almost the same percentage viral prevalence in the wild population as we see in our captive pythons, but with no disease at all. So it is entirely normal in the context of a wild python to have a quarter of the population positive for serpentivirus without any disease. And so it is entirely normal to have a perfectly healthy population of captive pythons that might have a 25% prevalence of virus. And so that's a complicated world to live in. That is especially complicated when you layer on limitations of different testing methodologies. And so, there's kind of several different paths that I can see that kind of the future of management of these viruses go, which is that you have these like specific tests looking for specific serpentivirus species. So one of the main problems is that when people talk about serpentivirus or nidovirus, they think they're talking about one species of virus. This is 7 genre, now potentially 8 genres, so 7 genuses, genre of virus with dozens and dozens of different species. And some of these seem to pretty consistently cause illness. Some of these pretty consistently seem to never cause illness. Some of these pretty consistently seem to only cause illness during stressful, aberrant, maybe during the breeding season, you'll get like the most mild respiratory infection. And so we really don't know what the recipe is between virus to species to secondary factors to the specific strain of virus. And so if people just talk about serpentivirus or nidovirus as if they're talking about one thing, you're already kind of off to a bad start. And so for some species, it might actually be kind of simple. Like ball pythons seem to really only, I shouldn't say they only get ball python nidovirus, but like we don't see the same diversity in ball pythons as we see in like green tree pythons. And so in the context of a ball python, you can use a pretty specialized test and feel safe that that test is probably saving you from a decent amount of risk that would come from stuff. But if you're looking at a species with a viral profile like the green tree pythons, where it's like almost half of these viruses are coming out of green tree pythons, you can be led astray by certain types of testing that is really not specialized in picking up a wide variety of viruses. And so there's limitations. Testing is a tool. And we hit the limitations of these tools when we see very diverse groups of pathogens. And this is a very diverse group of pathogens. So for some species that don't have that same diversity that's causing illness, it might be pretty easy to localize the problem. For other species, it might be more about managing the circumstance. And for other species, like the viral clade that we found in Burmese pythons in South Florida was very similar to a virus that I pulled out of a reticulated python from my original disease outbreak. And I elected not to euthanize that animal because it had a really weird virus in it. I had never seen disease. I'd used this animal for breeding multiple years, and none of the females that he was with were positive. And so I said, whatever's in this snake, if it was going to cause a problem, it would have by now. And so I let that animal exist in my display portion, my display building. And so that display building, I'm not breeding animals in there. So I'm okay with there being known positives in there if I manage it very carefully. And so, yeah, that animal has lived here for it's been 7 years now since the initial positive test, and it's never broken with a respiratory infection. I've never seen a sperm with one of these viruses break with respiratory infection. But, there's even within, so my blood python colony, my breeding adult. Yeah. So in the context of this display building, a positive test does not scare me because I'm not putting it in a situation where it could cause disease. In my biosecure building, zero virus. So if there's any level of anything, it would never make it into the building. And if I find that there's anything in that building, I don't care if it's symptomatic or not, that is a biosecure building with no serpentivirus in it. That is the basis of that building. That animal would never be accepted. So already I have two extremes. I have I don't really care what its status is, so long as it's not showing disease. And I only care about the status of that animal, regardless of if it's showing disease. And then I have my commercial building, which houses the adult breeders, that I call my virus neutral building. And the reason I call it virus neutral is that, so all the foundational animals of that building were neonates at the time of the original outbreak, never kept in the same area. So, and tested multiple times. So should be clean. They went into that building, virus clean, has not shown disease, kept that colony closed. And then 4 or 5 years later, in the middle of breeding season, I get a male that has the most slight respiratory infection. I take a sample from him, sequence it, and it's like a new subgenus of virus. So I can't lie to myself and say that that is a virus negative building, but I can also say that I can't find virus in that building unless there's a true exceptional set of circumstances. And then when I did, it was like a new subgenre of virus. Like it's like walking into your snake room and opening a ball python tub and there's a corn snake in it. Like it just, where did it come from? It came out of nowhere. So, if you can have a new subgenre of virus pop up in a 5-year closed colony where everything was tested multiple times going into it, this is a complicated world, So,
jeez, so that animal was negative for 5 years.
Yes, yep. And when it did detect it, it was a very divergent virus. So I know that it would not be detected on any other test other than the one that I used for it, essentially. Like it's too different. And so that would be missed. If I put that into a commercial lab, same with the retig, actually. I submitted that animal as well, and it did not come back as positive. So like there are entire genre of these viruses that I know are not being detected on many of these tests. Whether or not that matters maybe is less important. Like again, I don't think if a subgenre of virus can hide for 5 years in a healthy snake, and even when I do detect it, I get the most frail, broken up genome with very low copy cover. Like I'm not really scared of that. Like it doesn't, but in that same colony, when I see the slightest bit of clinical illness, so my virus neutral colony, I treat it with a priority on clinical disease. So I can't detect virus except for the most extreme situations. But when those extreme situations occur, I treat that very seriously. Because when you see clinical animals that are starting to show signs of respiratory infection, viral amplification goes way up. So a lot of one of the limitations on testing is sensitivity and specificity. So I've talked a lot about specificity, the range of virus that a given test can detect. But there's also sensitivity, which is how much virus needs to be in that sample to be detected. And the general trend we see is that the sicker the snake is, the more virus amplification, or the more virus is actually present in that animal. And the more viral replication you have, the more viral mutation you have. These viruses are not great at copying their genomes. And a lot of times they make billions of, human brains are not made to think of pathogen reproduction, but you might have the difference between a healthy positive snake and a sick positive snake might be literally billions of times more virus. And so if there's
Mm.
billions of times more virus in this sick animal than there is in this healthy animal, then you can start getting mutation. And when you start getting mutation, you now have a very dense colony of animals with a potentially mutant virus in an immunocompromised animal. And that is where I think we get these big mass collection die-offs that occur. So when my original outbreak occurred in 2018, I had two things that happened at the same time. The first thing was I brought in a small number of adult blood pythons into the colony. They went through a quarantine, but like a normal reptile keeper quarantine, like, a month, they look good. Okay, sweet. Move them into the rest of the building. At the same time, I moved the whole colony into a new building, my first outbuilding at the time. So we had two things occur at the same time. It's this universal mild stress pressure of being in a new environment with the addition of a new strain of virus. And that caused clinical disease, which then exploded. And that's when I started seeing a bunch of positive animals and a bunch of disease. And so there's a balance between stress on the animal, novel strains, and the specific, what the specific combination of virus and snake is. So now in this virus neutral colony where I can't find virus unless I have a reason to find it, I really pay attention to the reasons to find it. And that is when you get these mildly sick animals, then I start really treating them seriously, isolating them. And if it lasts anything longer than like a single shed cycle, so that's the most common time when you see it, is when an animal goes into shed, you'll see like it slightly dips their immune system and you'll get just the most hint of slightly thicker mucus than normal. Like you won't even miss it. It's not clinical. It's more just like if you look at the mouths of snakes every single day and then all of a sudden it goes into shed and you notice it's glossy in a different way. Like their gums become more of a matte texture than a glossy texture. And then and only then can I sometimes pull the most terrible genome out of that.
Interesting. So in your mind, is this something that we just sort of have to accept as something that is naturally occurring in these animals?
Yes. Yeah, absolutely. Yeah. So one of the signals that we are getting, and so there's dozens and dozens of species of these viruses. And so one of the questions we had is this a reflection of what we're seeing in wild populations, or is this an artifact of captivity? And the answer is probably both, really. But the reason that makes me think that the species of diversity of virus that we're seeing is a reflection of the species of viral diversity we see in the wild is how stable some of these viruses are. So the ball python nidovirus that I pull out of ball pythons today is like 100% identical than the ball python nidovirus that was discovered as the very first one 10 years ago. And so two things are occurring at the same time. One, we know these viruses mutate like crazy. And also, they seem to be very stable over a span of 10 years. And so how are both of those things occurring? Well, it is probably that these viruses are really optimized to grow in at least some of them are really optimized to grow in certain species. And what we are seeing in the viral diversity is an extension of that diversity seen in the wild pythons. Now, I can also directly conflict with that statement because we also have pretty good evidence of recombination, which is where viral genomes literally swap parts of their genome. And I can also detect that too. So like ball python nidovirus is very, very stable, but maybe green tree python nidovirus, all the diversity we're seeing is from this recombination and an artifact of captivity and keeping animals in the colony for a very long time. And so, we are definitely seeing feedback from the context in which we keep animals in captivity, but that feedback is coming from a wild source. And, that is, at this point, the very clear picture that is painted.
How many strains are there?
So taxonomy is just barely a real science. And taxonomy is humanity's best attempt at trying to classify the natural world and put it into boxes. But stuff doesn't exist in boxes. And so we really don't know. There's anyone's best guess. And the field of, so there's 26 different definitions of what a species is.
Okay.
Across science. So a species in a plant is different than a species in a snake is different than, so these are really fuzzy boxes that we're talking about. There's currently, I think, around 40 different recognized species of these virus. And what fits that criteria is 5% or more divergence in a certain portion of the genome. Past that point, we see a ton of diversity. It's basically it's not helpful to think of these as discrete species. It's almost like a sliding scale of diversity that occurs. I can say this virus is 5% divergent than this virus. So these are two very different species of virus, but there's one that's 3% from both of them. And so it's really just hard to say. I think the simplest way to put it is that there's a lot of diversity out there. Much of that diversity is probably captured by the concept of at least 30 different species. There's at least, 8 or 9 genre of these viruses at this point. So it's really broad, and it's not just snakes too. There's turtle viruses, there's lizard viruses, chameleon viruses, so and that's sort of where we pair the limitations of testing with the current state of our understanding of these viruses. So yes, a particular test might miss a divergent strain of virus, but if the understanding starts narrowing down to okay, these are actually the group of viruses that we need to be concerned about, it might, if you're trying to say, I want a colony that is entirely negative of virus, that is a very expensive thing to do. And you end up with a lot of answers and a lot of targets that might not matter. If we start backing up and saying, okay, well, let's really focus down on the ones that we know have a pattern of causing clinical disease, and that's the ones we're really strict about, then the limitations of some of these tests might be acceptable, But
so
yeah, as far as the specific strains, I think the latest paper I put up put almost 68 different published viruses that we have at this point. All of these are full genomes available online. So there's a huge diversity, and this information is not hidden.
So essentially it's a spectrum, and on one end of the spectrum there are snakes with what you would call Nido, and they're going to live a long, healthy life, and they may not transmit their disease. They may not show any symptoms, and that animal could live safely in your collection with proper protocols.
Yeah.
And then on the other end, there is the animal could show symptoms and end up, passing it on to other animals, and that animal could die and all your other animals could die.
Yeah. And potentially very quickly too. Like, so one of the experiments I did for my PhD was growing a lot of these viruses in culture in the lab. And that's how we've developed a lot of the information we do know about these viruses, like what sanitizers will kill them, how long they exist in the environment, stuff like that. Damn, I totally lost my train of thought on that. Hold on. Oh, yeah, can grow quite quickly. So one of the things we do is growth kinetics. And so essentially, I set up an experiment where a snake cell is infected with exactly one virion, one virus particle, and then seeing how long it takes for production of new virus to occur. And we basically see a cycle of growth is within 24 hours. And so it really can escalate very quickly in terms of bringing in an animal. It very quickly, from a stress event, developing clinical signs, because all it takes is one, a 24-hour cycle of growth, and then potentially spread that exponentially very quickly. And so it's scary and complicated, but like at the end of the day, the tools of it are pretty simple, which is just a long quarantine and good biosecurity. Like we see 6 snakes make 6 snakes. And so preventing the influence of 6 snakes is really the most cost-effective way of keeping a healthy colony long-term. Layering on top of that is testing and good, protocols and the risk appetite that you can live with. But the diversity of these viruses is so immense that any testing regimen is probably going to miss something. And even if it doesn't miss a Nidovirus or a Spentavirus, the list of unknown unknowns out there in the world is very, very high, especially in this group of animals that not a whole lot of veterinary research is being done for. We're finding new pathogens regularly in these animals. And so you really can spend a lot of money and a lot of effort and a lot of time testing and chasing down and trying to paint a particular picture. And at the end of the day, it's just one pathogen. And so for me, while I very much value the value of testing, like it's a very critical tool, I try to use that tool in the context of an environment of prioritizing health of the animal, because I know all the tools in the world are still, at the end of the day, have limitations. So health of the animal is a huge factor in terms of trying to figure out risk. And sometimes that can change too. So like I had a Papuan python that came in that came with a really weird virus.
This is Apodora?
Apodora, yeah. And for 6 months, I had this animal and it was completely asymptomatic. And I was really not sure what I was going to do with this thing, because all the other Apodora I had made it through my biosecurity and made it into the virus-free building. And so I was really on the fence of that. But I was like, it's very hard to get more of these males, especially. This animal's been here for a long time. I don't really think I'm that worried about it. And then all of a sudden it broke with a respiratory infection, and now I'm worried about it. But then I took that as an opportunity. Okay, well, now I have this animal. Let's try and grow its virus in the lab. And I was able to grow the virus in the lab, but it did not kill any of the cells I grew it on. So I couldn't do any experiments with it. And so I'm like, it's so impossible to make hard rules on any of this. amount of black boxes, like what people think are the resolution of our understanding on this is versus what it actually is. Like, there's a big gap there.
Yeah.
So even the things that I think I know about these viruses from growing them in the lab, is still only limited by the ones that I could grow and that caused death to cells such that I could quantify how much live virus was actually in a sample. I knew I could grow it. I didn't know how much of it I could grow because I couldn't titer it out based on it killing other cells. And so, yeah, it just, I am constantly having to remind myself that even what I think I know is limited to, of the 7 genre of virus, I've only been able to grow members from one of them. Within that one, only 3 out of the 5 that I grew worked in my experiments.
Mm.
And so, like, all of these things have, limitations. And it's important for people to kind of internalize that a little bit to sort of lighten the pressure as far as what a true negative animal might be. So, like, one of the challenges that we have is, and we've noticed this in both captive python populations as well as the Burmese pythons. In the Burmese python study, we had animals that were captured positive for a very specific strain of virus, then go negative, sometimes for like 3 or 4 months, and then pop a positive again with the exact same virus, which would hint that it's not just reinfection. Like, it is that animal's strain of virus that went below a threshold and is now popped back up. So they're like, you have intermittent shedding. So you have to actually have an animal shedding enough virus at the time of sampling. There has to be enough virus in that sample to meet the sensitivity of a specific test. And that specific strain of virus has to be within the specificity of the range of viruses that those tests can detect. And so for some species, that's very simple. Like a ball python is pretty simple. For other species like Morelia, it's harder. And so you really do, if your expectation is perfect, if your expectation is no virus, you really need to make sure you are not lying to yourself within the limitations of a given test.
Is that why you don't keep Morelia?
I mean,
aside from green tree pythons?
No, I really do miss my Morelia. So I actually don't keep a ton of Morelia outside of the green tree pythons, because the focus of my facility are species that are not regularly bred in captivity. So the commercial species that I have are really restricted to the blood pythons and Borneo pythons. And my diverse python colony, I really wanted to focus in on the species that were either, it's about half the world's python species, but I joke that the other half are the ones that are either completely unobtainable or only obtainable as multi-generational captive bred animals, where I really don't feel like I'm adding anything to the, to the pile there.
Sure.
Like, if I wanted any type of carpet python, it's going to come from a captive breeder. And so I didn't really feel like I could do as much, change there as I could with species that were not worked with commonly. Sure. So yeah, the reason I don't have Morelia is because I'm limited on space and because other people have done great work working with Morelia. So yeah.
Yeah.
But they are on my list. It just not couldn't prioritize them. But if I would go about setting up a Morelia colony because of the challenges that come with that, I would be buying neonate animals almost as close to time of hatching as is responsible to get.
Hmm. Do you have Tannenbar scrubs?
I don't. I had the opportunity to go for a male, but they've been so hard to find pairs of that I was worried about just getting stuck with them. But honestly, at this point, I'm just kind of at my capacity. I don't really have much of anything on my list that I'm really gunning hard for. But
Yeah.
I strongly considered the Tannenbars.
Sure. So I want to talk a little bit about disinfectants. What are the best disinfectants for tongs and water bowls and all that? Do you have a favorite, or are they all great? What is Dawn dish soap going to do? Is that going to kill the virus if you're scrubbing like down bowls with Dawn dish soap? Is that going to disinfect?
So I actually did not test Dawn dish soap specifically, but it actually should do pretty good. And the reason it should do pretty good is the reason all of these do pretty good, like almost any sanitizer does. And so there's many different types of viruses. You have DNA viruses and RNA viruses, and then you have enveloped viruses and non-enveloped viruses. And so we kind of lucked out in that this is a enveloped RNA virus. And so RNA itself is really quite fragile. And when a virus has an envelope, that envelope is a phospholipid bilayer. It's the same thing that forms the membrane of your cells. So actually, when it's leaving the cells, it actually kind of takes some of that membrane with it. And that forms the membrane of the virus, among other viral proteins. All you really have to do to destroy that virus is break that little layer of fat. And so soaps do that. A lot of things do that. And so the criteria to actually kill these viruses is not very hard. So alcohol-based sanitizers are totally fine. I use F10, quaternary ammonium. I use the combination of F10 and hand sanitizer or alcohol-based sanitizers. The only thing that didn't do amazing was peroxide. But like some pathogens, peroxide does really well for other stuff. And it's not, I should back that off. Peroxide did great. It just took longer. So most of the sanitizers completely knocked out the virus within a minute. For peroxide, there was a little bit of virus left after a minute, but after 3 minutes, it was totally gone. So one of the things that people don't consider is contact time. So it's not just enough to know, what type of sanitizer to use, but also to know how long it takes for those sanitizers to work. So like, one of the things I have in my exhibit building is in entering, I have that little scrub down room, and that has an automatic timer for 1 minute. So when I put on sanitizer, I sit there and I wait 1 minute. And that is a very long time. And so which sanitizer you're using is really only half the equation. It's really contact time for that sanitizer to work effectively. For serpentivirus, we found that most of those were within a minute. But if you are really concerned about it, you can, read the back of the packaging and it'll say what their recommended contact time is. But it's really not that tough. Most of these were knocked out within a minute, and most of that was like kind of under the manufacturer's contact time anyway. And then,
oh, sorry, go ahead.
Yeah, layered on top of that is also just how not stable they are in the environment. And so for the same reason, they're kind of fragile. All you have to really do is break up that lipid bilayer. They're maybe only lasting 5 to 10 days at room temperature. And if you are talking anything, a more high high heat, high humidity environment, it's probably even less than that. It's probably less than like 5 days. And even within that, we see a big drop after 24 hours of how many actual infectious virions are left in a sample at room temperature. And so the reason we see such a high prevalence in captive python colonies without necessarily seeing, if 25% of pythons out there are positive, we aren't seeing 25% of pythons out there dead with respiratory infection. We aren't seeing 25% of colonies out there suffering from disease. And that's, because these are honestly pretty manageable pathogens as a whole that have limitations in the environment that are pretty susceptible to most sanitizers and decent biosecurity. Yeah. So
So, I mean, one of my concerns is I have kids and my kids like to interact with my animals and everything. And kids, it's hard enough to get them to wash their hands after they go to the bathroom, so it's, that's why that was like one of my biggest concerns of, that's kind of what brought me to you, was having a concern of having a jungle carpet python that, was positive, and he had a negative result, which could mean he's negative, right? Or it could mean that he's negative right now.
Yeah. Yep. Yeah.
And if he is negative right now and he might be positive down the road, at least I know that this might be a strain that just causes a little bit of extra mucus during a shed cycle, and then it goes away, and he's for the most part healthy.
Yeah.
So kind of a fun fact that I learned recently, and I'm hoping you can back this up, is that if you're buying alcohol for disinfecting at Walmart or whatever, 70% is better at disinfecting than 90, right? Because you get more contact time because it doesn't evaporate as quickly.
Yes, yeah. That is the biggest limitation on alcohol, is that it evaporates really quickly. So yeah. Okay. Most of like hand sanitizers or something like that would be like 70%.
Okay. So something that is going to give you that longer contact time to destroy the virus.
Yes. Yeah, exactly. Yep. Yep. Yeah.
And what
alcohol is actually one of the ones that is like, it's really convenient. And so it's hard not to recommend it, but it can also evaporate really quick. Sometimes it can evaporate quicker than that contact time. And so that is what you need to keep in mind if you are using alcohol, which is why I use it not for everything. So F10 is what I do for most of my stuff, so how I manage the specific animals within the buildings is entirely different. And so in that biosecure building, it's sanitizers between each animal. In the breeding building, I have stuff in blocks. And so I'm sanitizing between blocks. Like I'm not going to sanitize between the animals if they're in the same breeding group. And then I have these breeding groups by project in a given rack, and I'm swapping gloves between racks. And so the hope is that I'm kind of creating these disease management units. And if I do have a positive animal, it's going to be limited to just that block of animals. And if it does break out of that block of animals, it's going to be limited to just that rack. But like I'm using alcohol between the breed groups because it evaporates pretty quickly and works pretty well. So my hands aren't like gooey, gunky. But, the tools I'm using F10 on because I trust that a little bit more, and I have the ability to give it a longer contact time because of what I'm doing in that instance. Like I'm not going to sit there and wait for my hands to dry a whole minute between each snake. So, but then, by the time I'm reaching a rack, I'm swapping out full gloves. So that should be a hard reset. And so, we talked a lot in COVID about like Swiss cheese models is like, every prevention you take has limitations. And so you can think of that as like a hole in a slice of Swiss cheese. But if you stack enough of them together, hopefully you don't have any holes for anything to pass through. And so I'm using alcohol between each breed group. Hopefully that'll stop it containing any problems there. I'm swapping gloves between each rack. So hopefully that'll stop a problem there. I am not working these buildings on the same day. So hopefully that'll stop a problem there. I'm not selling any animals out of that building. Like the animals I make available for sale are the most protected class of animal within that. And so all of these are made to contain a problem should one exist. And what made me make these changes was serpentivirus or nidovirus, but it's going to work just as well for other pathogens too. So like, because this one is so manageable, it's a great one to practice biosecurity with, because those habits that you build will extend out further. And so then you can live with the risk of knowing that you will never know every microbe in your animal, and there are going to be unknowns in there. But you are paying attention to the health of the animal and putting in guards in place to make sure if there is a problem, it's localized. But, as a snake keeper, if my intention is not breeding and keeping high densities of animals, then my bar might be, a little bit different. Like, if you have a collection of 10 snakes and that's it, like maybe you consider your unit that colony. And so then you aren't driving yourself crazy about, like, not gonna say don't wash your hands between animals. Like that's a good idea, but kids are going to be kids. And if you understand, your management unit, I almost look at individual colonies as having their own set microbiome. And so if you are aggregating those animals together as a unit with one microbiome, where you want to draw the line on biosecurity is kind of dependent on your risk profile within that,
Yeah. So you've got, I noticed kind of the flow during our visit was we started with the babies, right? So that's like your most biosecure. And then we went to your biosecure facility. Then we kind of went to your, what do you call
Exhibit.
it? Kind of your, you're just, we were all allowed in. We were able to go in and walk around, look at everything. We saw the Burmese python that doesn't have a tongue, which is still bizarre. And he doesn't have a tongue because he had tumors growing on his tongue, correct?
Yeah. So randomly, I noticed, this is probably about a year ago, he was flicking his tongue and there was a bump on it. And I didn't know what it was, but I caught it on slow motion video just to like convince myself that it wasn't there, or that it was there.
Yeah.
Then about 2 months later, I come back and the whole tongue is just like unrecognizable as a snake tongue. And so, we actually took him in, got him surgery, cut off his tongue with a laser. It like uses a laser to both cut and cauterize the wound at the same time. So I have this like crazy video of a Burmese python getting its tongue cut off by a laser.
Oh my gosh.
But he has since seems to be doing really well. Like that was over a year ago at this point, I think, that he had his surgery, and doesn't seem like anything has come back, but it was a new one. Snake tumor on the tongue was a new one. And I think I remember they were looking up, like trying to find if there was like a veterinary protocol for lasering off a tongue. But so I think that was the first one ever done. So the first laser Burmese python tongue.
For anybody that doesn't know, it's very bizarre having a snake look at you in the face and not flick their tongue. It's such a weird thing, because he was very intent on looking at me, but there was no tongue flick. So it was like something was missing. Part of their body language is missing.
Yeah, you really don't realize how many subtle cues come in from the tongue, just in terms of how intense he's licking, or how they're exploring the world. They really do share a lot. But it is very interesting now because he doesn't have the same, like, he knows there's rats in the building, but he will always assume there's rats in the building because his detection criteria is just way lower. So he's been very hard to read. He often comes defaulting to food response, and then you can kind of turn that off, and then at that point he's totally fine outside the cage. But yeah, it's the first few months was very confusing working that animal because he was an animal we had done outreach with for like a decade, and all of a sudden he became very hard to read.
That's too bad. Poor guy.
Yeah. Well, like I said, he still does great for outreach now. I don't think I'm legally allowed to because they're prohibited now. Like they're very restrictive on who can actually have contact with the animals. But yeah, he still does great when he's outside the cage. But when you're first opening that cage, he's very hard to read.
Yeah. Looking at him, it was like having my dog look at me. It didn't feel like a snake was looking at me because it's like that. Snakes communicate so much with their tongue. Like you can kind of get a vibe for what they're thinking or what they want to do next. So basically, if anybody is looking to disinfect 10% bleach, F10, H2O2, peroxide, you're good to go with all those. Peroxide is going to be a little bit longer contact time. What about like Clorox wipes? Are those just good for an all around kind of wiping surfaces down?
I, again, can't kind of make specific comments on that because I didn't specifically test that. And a lot of that has to do with like how we can actually test it. Like I have this liquid vial of virus. And so I like can't really wipe.
Squeeze the juice in.
Yeah. I don't really have a way to quantify the application of that in the same way that like I can make a 10% bleach solution. I can make an F10 dilution with it, so, but it is better than nothing. And all of these layers of biosecurity that you're doing are going to only tighten the holes in the Swiss cheese,
Yeah, that's right. Yeah. You made that point. And I remember that was kind of a big, point of contention, right? Was, masks don't protect and this and that. Right. But the idea is that, well, if you have a mask on and then you're maybe 6 feet away from somebody and you wash your hands, right? You're just layering that protection until you get to a point where you are safer than you would be if you had COVID and you were literally, right next to somebody with no mask on.
Yeah, exactly. And, COVID is, such a great example to use in the sense of coronavirus is a nidovirus. Like when we first discovered these viruses, we gave them the term nidovirus because that was the most broad thing we could describe them as.
Mm-hmm.
But nidovirus is actually not a very specific term. Like it's like calling a snake a reptile. Like, it doesn't actually help you classify that. So a coronavirus is also a nidovirus. And much of what we thought we knew originally about serpentivirus came from our understanding of coronavirus. And so for COVID, you had almost all the world's scientists working on one viral species. And think of how much is still kind of unknown, black box, subjective. Like there's a lot of, you had all of the world's virologists kind of working on one problem. And at the end of it, we still kind of have, kind of a fuzzy understanding of all the nuances that go into what made coronavirus really matter in some people and easily kind of dismissed in other people.
Mm-hmm.
And so that's the range we see in one virus with all the world's experts studying it. Here we have like 40 coronaviruses, 40 COVIDs going on in all of these reptiles, at least. And like 2 or 3 scientists working on it, maybe with no funding. And, so like.
And you're one of those 2 or 3.
Yeah, exactly. Yeah. Like you could give me $5 million in 50 years and like, I still don't know. I still think I would be able to sit here for 2 hours and explain to you all of the things that we still don't know. Like, one of the things that Eugene always stressed from his mentor is that numbers go infinitely this way and they go infinitely this way. And what I take that to mean is that not only do we see, at least in the context of pathogens, a huge variety of microbiome of microbes. Like, we really kid ourselves when we think this world is ours. Like, this is the microbes world, and we are just a microbe that's gotten a little carried away. So, but like, it is the microbes world. So diversity goes infinitely this way. There's a billion different bugs out there. And even when you just pick one, the diversity goes infinitely this way. We can't even classify them as discrete species easily without stepping over our toes a little bit in the process. Like, we can't make universal sets of rules that apply for everything for these without being able to find an exception in the mix. So, it is ample ground to drive yourself crazy. But what I always remind myself is that, herpetoculture is not new. These viruses are not new. And at the end of the day, the world is not burned to the ground yet. And so if that is the baseline, anything you add on top of that in terms of biosecurity and proper management is just going to benefit you. Like, we are it's not in a pathogen's best interest to wipe out a host population. The wild adapted version of these pathogens are oftentimes never causing disease, or only causing disease at like the end of an animal's life when it's really stressed out or in some sort of extreme condition. And so the mindset of virus is trying to kill host, and so I must kill virus, is itself kind of losing sight of the bigger picture. A lot of this is a balance. And so it's really challenging. And it's why I circle back to prioritizing disease and what a positive case means in the context of a given animal in a given colony.
So it's kind of like, like all those best practices and everything. It's kind of like when everybody was going back to the office and everybody was getting all sorts of bugs and illnesses and sicknesses, like right then, it's kind of, everybody just said, all right, we're back to normal. Therefore there is no more, COVID to be worried about. And, everybody's picking up all the other bugs, especially if you were isolated with your family, then you're growing your own strains of bugs, right? And just bringing them out into the public and spreading them. And so that's kind of where having your own colony, they will end up developing sort of their own defense against each other and their own microbiome. And I mean, that
is probably a reasonable understanding of it. Like, that is a reasonable take that will prevent you from driving yourself totally crazy. Like, my colony has a microbiome. I understand the risks that come with that microbiome and manage stuff in the context of that. But like, it's such a nuanced take that I'm trying to articulate here, because I want to make sure it doesn't come across that I'm saying that virus doesn't matter and that testing isn't important, because those are inaccurate. Like, testing is very important, but it is a tool that has limitations. And if you don't understand those limitations, you can come to a conclusion based on misunderstanding. And I don't want to say virus doesn't matter, because I've lost a colony of snakes to these viruses. And so there's absolutely contexts in which these viruses matter a whole bunch. But if you start from an absolutist mindset, you might end up in a place that is also based in misunderstanding. Like, it's not that these viruses don't matter. It's that these viruses are present and sometimes they cause disease. And it's really the disease that we see problems with. So a lot of these times when we see disease, it's often a co-infection with bacteria or even fungal sometimes. And so there's this synergistic effect between bacteria and circumstance of the animal and stress of the animal and virus. And so, like, all of this stuff coexists at the same time. And so trying to pull any of this out of isolation of that and try and base an understanding or worldview on it is just missing some part of the picture.
The importance of testing, do you think that there will ever be an at-home test for keepers that would at least tell you whether or not you have some form of serpentovirus in your collection? Do you think that that's something that you guys will ever have?
Almost certainly not. No. Yeah. So a lot of the at-home testing is, you sacrifice a lot. So this is, if the closest you might be able to get, if someone really, 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 really cared and had a bunch of money to spend for a bad investment, I think would be like, you might be able to get them down to, like, if you were to just target ball pythons, you might be able to make an at-home test that could capture the diversity seen in that. And even then, I wouldn't feel good about using it, but it would be better than nothing.
Okay. Just because, we were talking about COVID, and yeah. I remembered, when USPS was giving out the free COVID tests, right? And I only thought about that because right before I got on this call with you, I opened up my bathroom drawer and I saw one of the old boxes laying there. And it just made me think, like, oh, wouldn't that be cool if we had an at-home test?
No, so great example, because again, you have one virus.
Yeah.
And that test is made to pick that one virus. And even within that, you'll notice when you read the back of those tests that, like, the sensitivity is really only good if you were, like, pretty sick. Like, it has to be above that certain testing threshold. And so if you test too early, you're missing it. If you test too late, you're missing it. And most of them say that, a better test is a PCR test. So you really do sacrifice quality of answer in those type of tests. But the flip side of it is that they're cheaper and easier to run. And so if you're throwing numbers at it, you kind of can make up for it in the swing of things. Like, it's not as good of a test, but it's 5 times cheaper and instantaneous. And so kind of more effective in terms of actually managing the situation. So, unfortunately, the diversity we see is probably going to be too high. It's very expensive, and existing technologies in place are just better tools for it. So all of the molecular testing right now is PCR based, but there's 3, and I'm looking at adding 2 more categories within that of a specific PCR test. So a lot of the cheaper tests that we see are a qPCR or quantitative PCR, otherwise called a real-time PCR. And so,
what does that mean? Can you put that into, like, 3-year-old terms for me?
I guess I'll actually start first talking about conventional PCR. So conventional PCR is what UF does, and it's more expensive. And I think UF is one of the only people who does offer it. And as you know, it's hard to submit stuff to UF. So
depends on who you know.
Exactly. Yeah. Exactly. Yeah. So, the best test is also the hardest to access, and it's expensive, and it's potentially not cost effective for many people in many circumstances.
Yeah.
But this test that we use is a conventional PCR. And so what we do for that is we have specific primers that are made to, we have genome of virus, primer. We have two primers, a forward primer and a reverse primer, and we amplify what's between the two. Because these primers are made to detect the most amount of stuff possible, they match with a bunch of other stuff too that they're not really supposed to. And so if all we went off of was something amplified, we would end up with a wrong conclusion, because lots of stuff is amplified and it's not necessarily what we're looking for. So then what we do is we take that PCR product and we run it out on a gel, so it's separated by size. And we know that what we're looking for between these two primers is only a certain length. And so now we're filtering it by size, and only things that are the right size are suspected positive. So now we have amplification from our primers at the right size, and we think that is probably a positive. Many places that run PCR conventionally will stop right there. What we do is take that suspected positive and submit it for sequencing to try and determine what exactly virus it is in this case, but also to make sure that it actually is virus. So a lot of times these probes will bind with, or the primers will bind with a bunch of different stuff. And that happens to be the exact same length as the fragment of virus that we were looking to amplify. So, for example, if you have a really high mycoplasma positive snake with no nidovirus, our test will sometimes make that animal look like it's going to be positive until we sequence it and see that it's mycoplasma. Sometimes we'll get like a band that looks like it's positive and it's snake genome. So,
wow.
One of the limitations of our test is that in order to pick up as much viruses as we can pick up, we also pick up stuff that is not virus. And so to filter that out, we have to sequence it, and that costs money. But the result we get out of that is not just, is it positive or negative, but what exactly it is positive or negative for.
So that sounds like it minimizes the likelihood of a false positive or false negative?
It would minimize the chances of a false positive.
Okay.
One of the limitations of this test is that it makes it a little bit less sensitive because it is looking for a broad category of things, and it's a little bit loosey-goosey on what it can attach to, especially if we change the parameters on the test so that it one of the things we can mess with is temperature. So people think that science is science, but oftentimes science is more of an art. And so I have these primers and I'm submitting them to a thermocyclic conditions that are changing. And I can change some of those parameters. So like if I want to lower one of these temperatures, it'll make those primers more likely to bind with stuff that isn't 100% match. So if I'm going fishing for something really weird, I'll just crank that temperature low. But the expense of that comes of it's going to bind with more stuff that I'm not looking for, that I need to filter out by sequencing. But if you have something that is really divergent and the primer is only just barely kind of like it, it might not amplify as well as a different type of test that was really specified in on that particular virus. So what I've talked about so far is conventional PCR. It is more expensive. It's very time consuming. If I have to submit something for sequencing, it has 2 days that it needs to go there to the sequencer. Then I have to come back, interpret those results. Sometimes they're bad, so it sends them out again. It is time intensive that adds, limitations to some applications for testing. But when I was making my biosecure Python colony, that is the test that I used because I had really diverse Pythons. And so I want to use a test that's going to pick up anything in there, even that kind of looks like a serpentivirus. So that is what UF specializes in with this type of testing. Is you kind of have, it's useful to kind of think of known knowns, known unknowns, and unknown unknowns. And so UF testing specializes in known knowns as well as known unknowns, which is that we have all this diversity of pathogens, and based upon this diversity of pathogens, let's make a primer set that should pick up all of them. And so then in the future, because they can pick up all of this existing diversity, when you have these weird strains, they're likely to be picked up. And so using the diversity we see in our known knowns, we can make a test that is very likely to pick up related unknown viruses. So major pro for this type of test. And it's like, if I don't know what I'm looking for, this is the only type of test that I would truly trust. So that is conventional PCR. What most testing labs offer is qPCR, or quantitative PCR. So quantitative PCR does not have the same sequencing step. It does not have the same gel step. All it does is, so even within qPCR, there's two different types. The cheapest test that you will find available is typically uses a dye called fluorescent indicator that is called, like I said, Sabine green. And that is just showing how much amplification is occurring in a reaction, but not checking what is amplified and not checking what it is that is amplified past that point. And so if you have something that has a very limited diversity that you can make a test that is very specific for, that's fine, because you only know it's detecting what you care about. If you're trying to use our strategy, which is picking up the most amount of diversity possible, and you try and put on this test, you're going to end up with a bunch of false positives and no way to know that they are false positives because you're not checking to see what is actually being amplified. So one of the ways to layer in complexity to that is to add in, instead of using this general fluorescence that's just going based off of how much amplification is occurring, is to use a probe-based qPCR. So now we have our primers, and then in the middle, we're going to have a very specific probe that, if amplification occurs, will fluoresce. And so now I need 3 spots in the genome that are the same for everything I'm looking for: forward probe, forward primer, reverse primer, and a probe. But then I at least know that what I'm detecting is not non-target byproduct from the test. So by adding in that probe, I am reducing the specificity of the amount of pathogens it can detect, but I'm actually increasing the sensitivity oftentimes, because I'm only looking for one specific thing and I'm looking for it very closely. That type of test can detect that pretty well. So if you have a known, and that is all you are worried about, and you know a specific qPCR test can detect that known, then that is a cheaper test to run and potentially a more sensitive test. But if you don't know that test can detect what it is you actually care about, and a lot of them cannot because of the limitations of the technology, that they need to bind very specifically to not amplify stuff you aren't looking for, and there's just not enough spots in those genomes that are very specific between all of them. So it is almost certain that a qPCR test is only capable of detecting a subset of viruses that match really well with the primers and probe. Or if you make a set of primers that's good enough to detect a wide diversity, you run the risk of a false positive because there's not an easy way to differentiate something those primers are matching to by mistake. So all of these are limited. Like UF is telling you about the presence of a much wider diversity of viruses, but you're potentially losing out on a little bit of sensitivity, and you're definitely losing out on turnaround time, versus a qPCR is going to be a cheaper test to run. It's going to be quicker to get those results to you, but you're not going to have the same resolution in your answer unless you make even more intense tests. And so one of the ways to get around that is to like, okay, well, maybe I make a probe for all the ball python viruses, and a probe for this group of green tree python viruses, and a probe for this other group of green tree python viruses. And so now I'm testing for 5 things at the same time I'm testing for 1 thing. So like, all of these are tools. All of these tools have limitations. All of these tools have strategies to mitigate those limitations. And all of that requires an understanding that we don't necessarily have at this point. And so, a positive test is frequently a very helpful bit of information. A negative test can very much mislead you if you put too much faith into what that negative test actually means, because there are many, many reasons a test can be negative beyond just not being positive. But again, I really don't want to stress that testing doesn't matter. That's not what I said in that sentence. Like it
Yeah, it's just so. Okay, so all right, Dr. Steven Tillis, science artist. Man, yeah, that's super involved. And so you're basically, you have to start out casting the widest net possible, and then try to see if you can get something to fit into a box, and then try to find smaller and smaller boxes to fit that into.
Yeah, exactly. Again, if your question is, does my snake have a serpentovirus?
Yeah.
That is how I would do that. If I'm asking, does my ball python have ball python nidovirus? That is a different question. And so a different tool might be an acceptable risk profile to use to answer that question.
Okay. All right. So what about potential treatments? Are there any antivirals? Is there anything that is being worked on? Do you see one day being able to take your snake to the vet because it has an RI and, it tests positive for Serpento, and now we're going to give it an injection and it's going to cure the virus? Is that something that could potentially happen?
We are very hopeful. There's a couple of different levels to look at that I view as hopeful. So we did do some, Justin Julander did a lot of work with so we did an in vitro paper, and he did work on that for antivirals, and shows some promise that there's some antivirals that might end up working okay. But currently we're not doing any experiments with that. One of the things we are investigating is heat treatment, and we are hopeful about that, but there are a lot of things that make us very nervous about that, and a lot of things that I have described up until this point in terms of testing that are making, we really, really want to make sure that we are putting out good information for people. And part of that requires building really good tests. And so that is the stage that we are at right now, is still collecting an understanding of what, some options might be for some of these animals. But I definitely see a future where there is probably room for discussion on interventions that can be used for clinically suffering animals that hopefully will show some promise in terms of truly getting rid of virus. But there's several very tough things that come into play when we're looking at this question, because one of the challenges we see is that these viruses are intermittently shed. That is what we saw in the Burmese python study. That is what we see in captive colonies, is a snake will be positive and then be negative for a long time and then be positive with the exact same thing again. My blood python colony that was closed for 5 years that popped up with a new genus or subgenre of virus. Like, these viruses are really good at hiding. And so it is really hard to make sure I'm very confident in saying a treatment is working and not a animal has just quit shedding virus temporarily. And it might not even have anything to do with the treatment. So this is definitely something we are looking into, but I don't think the answers are going to be as simple as people will want them to be. And ultimately, I feel like it's going to be a situation that is really only reserved for animals with no other choice of intervention. Like, there's a lot of these animals that only get the most minimal clinical signs during shed. Like, I worry that people get too freaked out and animals might end up, paying a cost from improper medical interventions that are, frankly dangerous for the animal.
So I we've talked privately about some of the stuff, and I know that you don't want to go public with this because you don't potentially want people doing things that could harm their animals, etc. And so you had briefly touched on the heat treatment, and I just wanted to give you the opportunity to tell people, don't do these things yet. There's not enough research. It's not a good idea to do it yet.
Absolutely. Yeah. So it took me 4 years to be able to develop the resolution of saying which sanitizers work. Like, science is a slow process with checks and balances, and for good reason, and all the above. So, there's a lot of very big question marks right now that we really want to make sure we have answered, that are going to be problems if people take matters into their own hands. And so one of these things that we talked about is intermittent shedding, making sure that a treatment is actually doing something and not just we're tricking ourselves into thinking it's doing something because of the limitations of testing and what comes from that.
Yeah.
The other reason is that a lot of these are secondary to bacterial infections and stuff like that. And so you might actually have to treat more than one thing at a time.
So you could potentially kill your animal with another disease.
Absolutely. Yeah. Like if you do even more than a few, yeah.
Okay.
So
yeah.
And then the other limiting factor is that I can grow. So one of the limiting factors is viral mutation. If it is not done 100%, so there's not 100% of virus gone, it means whatever makes it out of the end of that is resistant to treatment, and that could snowball like almost immediately. And so then anything that might have worked now immediately doesn't work. And to take that even one step further is that I can grow these viruses on a lot of different cell lines, including multiple different species of mammalian cell lines. So whatever is keeping us from catching serpentovirus from our snakes is not limited to whatever the cellular receptor is. Like, whatever the cellular receptor is that allows entry into the cell is the same one in us as it is for the snakes. So we don't really know what the limitations are that prevent viruses from jumping between any number of different organisms. But one of those things seems to be the difference in body temperature. And so if you're starting to select for stuff that can survive warmer temperatures, like there's the potential for a very slippery slope very quickly. And so we really want to make sure we have a resolution on these answers, or else you're going to hit problems.
So serpentovirus can attack human cells.
I cannot say that. We did not grow it on human cell lines, but I can grow it on cat cell lines and dog cell lines. But I should even step backwards from that, because I can infect those cell lines, but it can't sustain an infection on those cell lines.
Okay.
And so again, there's nuance going on here. So even just like, it's not enough for a virus to be able to infect the cell. There's many other processes that go on that actually lead to an eventual infection, but we don't know what those are. And when we start mutating stuff, we might find an answer that is not good.
Yeah. So snake keepers, don't fuck this up for us.
Yeah, like for real. Yeah. Yeah. And it's something that I'm working very heavily on. I have come up with a new category of tests. So I've described to you the 2 PCR techniques that are available for commercial testing, but there's 2 other technologies that I'm working with that I think will actually be able to. So I mentioned the pros and cons of each of these 2 techniques. And there's another technology that is kind of now available that I just got access to, that is kind of a hybridization of the 2, that I think will be able to make sure that the data we are collecting is as good as can be for really understanding what is going on in these animals.
Okay.
But
and
yeah, that started with, understanding which viruses we are looking for. And now that we know we're only looking for these viruses, then I can make a test that is really specific to them. And then I can go and make sure that it's not just low because it doesn't bind to the primers. It's low because it's not there, because I made primers for that virus and I have a test for that virus and I cannot find it. And so these are the roadblocks. Yeah. The limitations of the tools are what we have to work within to gain a useful answer. And right now we're just not at that resolution. We don't have that. We just don't have that available.
Yeah, it's I mean, the whole premise of The Last of Us is that like the cordyceps fungus like can't grow in the human body, right? But the environment's getting hotter, so it mutates, and then it can grow in the human body, and it takes over your body, and you become a fungus zombie, right?
Yeah.
That's mutation, and that's so you want to avoid that, and so that's that takes very careful scientific work. And it's just not something that people should be playing around with at home.
Yeah, like one of 3 things might happen if you are experimenting on your own. You will kill the animal, you will mutate the virus, or you will not be sure if you have killed the virus.
All 3 of those suck.
Yeah. Yeah. But it is a question we're trying to answer, because how could you not? Yeah.
Yeah, absolutely. Yeah, it's it's all so nuanced, and it's a very delicate topic, and I mean, it's the same reason why you don't share your, you don't get antibiotics, and then when your friend has a sinus infection, you say, yeah, there's only 6 days here worth, but you'll be fine, just take them,
Yeah.
and then what's left behind after day 6?
Yeah, exactly. Yeah.
So, well, that's cool. Yeah, I wanted to really drive that point home.
Yeah,
Because
I really do want to thank the community for their patience on this, and circle back that all of my PhD research, and maybe not all of it, but a large portion was funded from Carpet Fest community donations.
that is incredible.
It really is. Yeah, it's been truly a privilege, like, to work on a project that works, how you think science should work, where like you have a group of people who like really care about a problem enough to actually collect funds to try and answer it, donate those, put that money towards a cause towards answering it, and then actually come back with answers for people in terms of, so all of what we know has stemmed from funding from the community. And so it is definitely my goal to feed back the right information into this community, because we wouldn't know anything without it. So
that's incredible. It's crowdsourced science.
It really is. And it really is a privilege. Like, there's many people in their research do not get, they work with stuff that like interests them, but it's not like, I got to work with my viruses. Like, that's a very cool privilege. And to be able to have this community that I grew up in fund the ability to answer some of these questions that, I remember after my outbreak having so many unknown questions. Like, it just didn't make sense that clearly multiple things were going on at the same time, that I was having a huge viral prevalence and viral diversity that I never had any signal of knowing that I might have until I had one of them start showing disease. And so then I'm left at the time with this understanding of like, did these 9 strains come in from this one that was sick? And that doesn't really make sense, because like, I don't even keep this positive green tree python in the same room as the blood pythons. And we just didn't know at the time of if that represented one virus making it in, exploding at the diversity within the colony, and then, causing problems from there. But now we understand that no, it was like 9 separate infection events over the span of however many species and years that I was keeping them. And only one of them ended up mattering to cause disease, or 1 or 2 of them. And so like, it's, I don't know. It has been scary and confusing and fascinating at times to be right at the forefront of understanding from the very beginning of when it started, of like, we truly have no clue how these infection events occur, to now being at the point of like, oh my gosh, there's huge viral diversity. We can find it in these wild animals. We can find it in vipers and chameleons and skinks and whatever. And all of these seem to have very clear different evolutionary histories. And 10 years later, a lot of these genomes are very stable. And so they're like, kind of seem to move through time as a species that doesn't change a whole lot. But also we see a huge amount of this diversity, and trying to square all of this conflicting information and not drive yourself absolutely insane in the process. Like, there's a lot of fear that comes from misunderstanding, especially around these pathogens. But like, I certainly feel a lot less scared of them now than I did in 2018. And a lot of that comes from the understanding that what we are seeing is just kind of an extension of natural processes put into unnatural circumstances. And it's up to us to manage those unnatural circumstances to keep a microbiome of the animal in healthy bounds. So like, one of the things that really drove home that point to me is part of my research was establishing a bunch of these different reptile cell lines to be able to see what diversity of animal serpentivirus can potentially infect, which is how I say, I can grow it on, or I can infect cat and dog and monkey cells and stuff like that. I also tried it on a bunch of different reptile cell lines, but there's not very many reptile cell lines available. So almost all of these were ones that we established. And so these are reptile cell lines that we established under the most, tight sanitary conditions to establish a completely clean cell culture to divide these cells into a cell line that we can freeze back. But it became very clear to me that even when you take the most sterile sample out of the most, a specific organ systems, that all I'm growing is like the heart cell of a Burmese python, you will still grow part of its microbiome in that.
Wow.
So one of the isolates that we had of serpentivirus wasn't grown intentionally. It was grown because a cell line was made of the animal. And then we found out later that animal happened to have serpentivirus. And now we have the ability, now I can take that serpentivirus and grow it on other cells in the lab. So like, it is not possible to culture any part of an organism without also culturing the microbiome that kind of exists within that. And if you scale out that concept, like, not only can I not grow a heart without accidentally growing serpentivirus sometimes without even knowing it, extend that out to a whole animal and the whole microbiome of that animal. And like, we kid ourselves when we tell ourselves that like these animals are sterile. Like, serpentivirus is just the most recent known, but like, we get unknown unknowns all the time. Like, we really do. So I think it is helpful for people to look at an animal's microbiome as really just more of an extension of the animal, and understanding that you can't take an animal out of the wild without also taking its microbiome with it. And that is ultimately where these things come from.
Yeah. It was your collection getting infected in 2018. Looking back at it now, that altered the course of your entire life, right?
Oh, absolutely. Yeah. Well, so, and this is what's funny is like, I didn't even know that. So yes, it absolutely altered my life. I had no interest in pathogens at all whatsoever. Like, could not. I like reptiles, and having to do homework on pathogens, and it's so confusing, and there's so many of them, and how are you supposed to remember them all? And like, and it's boring. It's not fun. Like, I don't know. It's what got me into it was seeing a snake on a branch in the wild and wanting to extend that feeling that I could feel every day. And so to have that spiral into a point where like that was threatened by a microbe, now it became important for me to realize, what did I do wrong? What happened? And so what really started as kind of my own curiosity from what truly did interest me has grown into really an understanding and appreciation that I feel goes, fairly deep, that can include an appreciation for microbiomes and pathogens and understanding that stuff is complex and all of that. But yeah, it absolutely changed my life. But what's funny is now that I have the knowledge that I have, I can see that 2018 was not new. Like, this is something that's been haunting the industry forever, including in my role in it. Like, blood pythons are known for being sensitive for respiratory infection. And that was just a thing that people understood as part of keeping them. And yeah, we could manage ventilation and temperatures and humidity, and that would reduce the chances. And then we come to find out like 10 years later that like, oh yeah, and actually a virus was responsible for what was actually going on causatively, but it doesn't negate the fact that all of these things that we were doing to help prevent sickness from occurring were kind of baked into the husbandry of the animals themselves. Like, same thing with green tree pythons. They are known for being sensitive for respiratory infection, and then come to find out, oh, they actually have a very high viral prevalence and diversity. And so it's like, nothing changed in 2018 other than now we had the ability to ask the question of what is this? And the answer was serpentivirus. But in terms of its effect on the industry, I don't think it's changed. I think it's always been there, always will be there. I think you can watch it move through species and colonies through the anecdotes that people tell of like, oh yeah, blood pythons are really sensitive to humidity and get respiratory infection easily. Which is to say, oh yeah, if you don't keep your parameters tight on blood pythons, they can stress out a little bit, and then a normal part of their microbiome can progress to a point where it causes disease. And if you don't treat that appropriately and isolate it in an immunocompromised animal, it can mutate into something that can threaten the whole colony. And also, it might be a weird strain that might be missed easily on tests. So good luck. So, again, I've done the best I can to drive myself crazy with it, to try and answer all of these questions, and ultimately just come to the fact that, like, we will never really have that tight of resolution on the understanding of it, but we still have really great tools available for us for managing this stuff. We have really good information that has now become available to understand the limitations of these. And, you can very easily live within those limitations and have a happy, healthy colony that will last many years, and you will enjoy herpetoculture, and you won't have to hit your head against the wall for 4 years trying to understand what causes some snakes to get mucousy sometimes, occasionally.
Yeah. And so as terrible as it was that you got that outbreak in your collection, like it seems like so much good has come from it.
I'd like to think so, but that's also, I'd like to think who I am, trying to make the most of a situation. Like, I've always tried to accept circumstance, and within that context, try to navigate to what's the best future within that. So, yeah, it really is. I remember the first blood python that I ever had that died of respiratory infection for unknown causes, like 15 years ago, because that's what blood pythons did. And I was so upset, and I ended up actually skinning that animal and preserving the skin. And I had it up on my wall for 15 years. And right as I am writing my dissertation, I look up and I see it and like realize, oh my gosh, this snake died of serpentivirus 10 years before we had a name for it. Like, this is, it really, at that point, it didn't feel like something in 2018 changed my life. It felt like the continuation of a story that I had been experiencing since 2009.
Yeah. I mean, so how did you go from getting the virus or becoming aware of the virus in your collection to now working at the lab and at UF?
In terms of like, well, so at the time, my advisor was Dr. Robert Ossoboff, or Oz, and he was on one of the first. So there were two papers that were published around the same time describing the first nidoviruses. One was in a Burmese, in an Indian python, and the other was in ball pythons. And so he was the author on the ball python one. He had just started a lab at UF, and I happened to meet him around the same time as he was starting his lab, right when I was having this viral outbreak occur. And so we basically had a bunch of opportunity for projects right there, a good start for him to be able to welcome someone into his lab. And then one of the other benefits that I had is I'd also been through building databases for other snake colonies. I had a lot of data already. I basically already had a master's project. And so it was very easy to start at the vet school and graduate school because I basically already had a project ready to go. And from that, we were able to add in all the Nidovirus or pentavirus stuff and turn it into a Ph.D. And so that really got my foot in the door there now. Now that I have graduated, I decided to go back to the lab as sort of experimenting in future directions for it. So experimenting with some of these various new testing technologies and stuff like that to be able to offer appropriate testing in a potentially hopefully easier way for people to access, but also just to explore if some of these other technologies are even valid methods for testing.
Yeah, that's it's a terrible thing that couldn't have happened to a better person. I mean, it's
yeah, it was. I mean, it was truly hard at that point. Like I remember, euthanizing my life's work. Like that, it was a loss that I still feel. Like there were animals in there that were irreplaceable, obviously emotionally, but even just outright. Like they're just, yeah, it was a very tough time. And like I said, the hardest part was determining if it was the right choice to euthanize healthy animals, even though they were virus positive. And like, now at this point, like I said, I just, knowing what I know now, I think I would have handled things a little differently, but knowing that it was the best understanding we had at the time. And, some people are still going to be, that is their criteria for what they would consider a safe, acceptable risk. But like the hardest one was a green tree python. And it was a green tree python that I had had for over a decade that had laid multiple clutches for me, never exhibited any signs at all, but was positive. And that was probably the hardest one. Cause yeah, it just, but I was no virus was my limit for that decision making. And so, that animal now, especially knowing what we know about green tree pythons, which is that they are loaded with a lot of virus that, I mean, that animal had lived in a colony for 10 years and never passed along its virus to any of the animals that I had bred it with. And so like, I would like there to be a place for that animal to exist that isn't euthanasia.
Yeah. If somebody gets testing done through UF, and it is probably the most costly serpentovirus testing that you can have done, is all of that cost going towards the testing, or is any of it going towards funding the lab overall and this whole project?
Yeah, actually, so that is a interesting benefit that we have, is that a lot of our funding, because it is from people testing, provides funding that is kind of independent from a lot of these, like, otherwise we'd have to go chasing down grants and stuff like that. So we actually are able to do a lot of research based on funds that come in from the lab, but it also is very costly to run some of these tests. Like it's not, to run an appropriate test with an appropriate extraction protocol, appropriate positive controls and negative controls. All of this takes, and especially if you have to send out for sequencing, all that actually does take quite a bit of money. So it's an expensive test, but it's also expensive because it's expensive to run the tests. But it is able to provide a fair bit of funding that we're able to use for various research and stuff like that. So it actually really does feed back into the system.
I think that's fantastic.
Right? It really is cool. Yeah. At this point, I think almost most of our projects are coming out of funds from that. But like university funding is 10 times more complicated than I have made any attempt to try and understand. So yeah.
So how does that work when there's funding coming from like Carpet Fest and everything does that go to the college? Like, how does that happen?
Yeah. So that was a smidge different. That was a smidge different because I was a graduate student at the time. And so we actually had like a specific project that we were, again, like there's layers of university bureaucracy that I've like done my best to not try and have to figure out. But there was a specific project that was associated with my time there as a PhD student. So the funds were like specifically able to go to that. Now that I'm not a PhD student, we have more of like a general lab research that we're working under. But a lot of it, I don't know. It gets really messy, which is how you can kind of get into all of the complaints that people have about academia and universities and stuff like that. Like, you know, those don't come from nowhere. There are challenges that come from working in a university setting, but also a ton of benefits. And, you know, we really have so many tools available to us that we can pursue very interesting projects.
Yeah.
But it's also, you know, not a commercial lab, but a lab that is subject to commercial forces that exist in a research setting. So it's, you know, in and of itself, it is an interesting place to exist in academia that I am appreciating.
Yeah. You said that you're working on ways to make the testing more accessible for the general public, so you could just go and get the tests yourself. And do you think that would actually make more funds come into the lab as well, so that way you could do more research?
Yeah, quite potentially. Quite potentially. That is what we're hoping. I feel that's going to be a little bit further off of a goal. Like I said, for right now, what I'm trying to do is explore various testing methodologies that we can use that'll start reducing the expense of our, what I consider the gold standard of tests, but not sacrificing the quality of answer that you'd get with a cheaper testing alternative. I am very hopeful that there's going to be a pathway in there that will provide a good balance of people that will make it easier for us to be able to accept stuff from more people too, you know? Like, honestly, right now, I don't know that we could, it is incredible to have a position where I am looking at hundreds of different pathogens from hundreds of different animals from dozens of different sample types. It really does give you an appreciation for just how many microbes there are out there. So we are trying to provide good testing within that sort of makes everyone's life easier and quicker, that doesn't sacrifice on quality of answer.
Yeah. Can a mother pass virus to a baby snake?
Does not seem so. Okay. So that is why I am willing to accept what I'm calling my virus neutral colony of adult breeders, is that we don't have any evidence to suggest that they're being passed vertically. So even if you had a collection of positive animals, if you are pulling the eggs, and especially if you're like, I go as far as to wipe them down with sanitizer, but that's probably way overkill. But really? Yeah, but there's no evidence that we have of vertical transmission. And so you should be pretty safe of having an adult colony that has a status that is unknown, and, you know, the offspring from that should be just as fine as ones coming from a negative colony. There are two things that give me pause to say that, like, you're totally fine. The first is that while we consider these respiratory pathogens and we find virus occurring in oral and respiratory tissues, they have found viral RNA in gametes, and I think even some of the oviducts. But there doesn't, when you look at the histology of it, it doesn't seem like viral replication is really occurring there. And we, based on testing so far, we haven't really seen any examples of vertical transmission. But, you know, I get nervous of saying it's impossible, but we certainly have absolutely no shred of evidence to suggest that that is the case. And it doesn't really fit with what we're seeing. But the one thing that does make me hesitate just a little is that in the original disease outbreak where I tested everything in my colony, I had 2 ball pythons that tested positive. Only 2 out of, like, I don't know, 30 or 40. And one was a mom, a dam, and the other was an offspring from that year that was not kept in that same room. So, but that's a sample size of 1. And I have plenty of explanations that could be not vertical transmission as to why that would be the case. But whenever I say, you know, I don't want to say vertical transmission is impossible, because I never, yeah, that is always in the back of my head of but that one ball python. But, like, in terms of mechanistically, like, that isn't really what seems to occur. Like, these are respiratory pathogens, and the disease transmission that we see seems to be a factor of exposure time. Like, baby neonate animals are a lot safer than adult animals, and a lot of the prevalence of virus seems to be related to the age of the animal, which means it's kind of a factor of exposure time. So it seems like extended time living in a colony of other animals is how this virus is passed along, not that we can see evidence of vertical transmission. But there's also circumstances that would be very hard to suss out between vertical transmission or not. Like if you had a boa and it gave birth in the cage, because there are boa nidoviruses, by the way. Not many of them, but they are there. But if you had a boa that gave birth in the cage, I think you would have a very hard way of not preventing contamination that would occur. Like, we are lucky with pythons that pythons lay eggs, because we have a 2-month gap between the mom and the baby hatching out. But if you're doing maternal incubation and that mom is sitting there coiled on those eggs, breathing on them as they're hatching out, like, yeah, that's probably
stressed out, not eating.
Yeah. Like, at that point, like, you might have viral positive offspring, and I don't know you'd have any way of determining that wasn't because she was sitting there incubating them versus, like, actually passing it along through the egg. But it seems very unlikely that there's any evidence of vertical transmission. Or it seems very likely that there's vertical transmission, and we have no evidence for it. And just similar pathogens just don't really act that way. Like, these are really respiratory focused pathogens. But, you know, every time I think I can make a firm statement about anything, I have to remind myself that my understanding is coming from a limited window, and, you know, never say never. But again, like, if that is the case, then, like, what are you going to do at that point? Now, at that point, there's nothing you can do about it. Like, you can't even break a pathogen generationally. So, you know, and there is stuff that is passed down in that way for sure. But thankfully, it does not seem likely that serpentivirus is going to be one of those. And so, for me, again, like, if you gave me, like, a very limited budget and said, how are you going to make a safe snake colony without any testing whatsoever, I would put things through very long quarantines in a very separate area. And I would keep my for sale offspring in a very separate area than the adults. Like, sometimes the cheapest intervention are walls.
Just having separate rooms that you go to.
Yeah. Yep.
And so you mentioned the boa nido. Can the boa nido jump to pythons, and can python nido jump to boas? And can veiled chameleon nido jump to a python or a blue-tongued skink nido?
So we don't think so most of the time. But we cannot say that absolutely, because that chameleon nido is sitting right in the middle of a group of python ones. So like at some point in the evolutionary history of these viruses, there was spillover between lizards and snakes. So it's like these things can happen, but they happen on like the event scale of evolutionary time. It doesn't seem like they happen short term. It seems like a lot of these viruses have pretty limited host ranges, of saying that like most of the python viruses we only see in pythons, most of the boa viruses we only see in boas, and they seem to be pretty localized within their spot on the phylogenetic tree. Like all of the viper viruses that we just put out, those are all a new genus, and that seems to be the only thing in there. So as a general rule, they seem to be pretty host specific, but there is evidence in the evolutionary history that that isn't the case 100% of the time. So like spillover probably does happen, but when it does, it represents a major branch in the evolution of these viruses, if that makes sense.
Okay.
But like we know it happens because it's there in the genomes. Like there's chameleon viruses and python viruses that at one point shared a common ancestor sooner than some python viruses shared a common ancestor with other python viruses.
That's wild.
Yeah.
Okay. Well, I think we've said everything we can say on Nido and serpentovirus. I mean, just outside of what is a good quarantine to you? Keeping on paper in, tubs that are separate, totally separate from your collection. You service them last, and how long does that quarantine last?
Yeah, so I actually have a few stages to it. So
Okay.
the first stage is like when something first comes in, and that'll be like when it first comes in, I will typically soak an animal in water just because I never trust anyone to be free of mites. They'll go on like a paper substrate. I might spray it with propenamide depending on how sensitive that species is towards it. That's when I would do initial testing. And once an animal passes that very first round, then I will actually move it into true quarantine. So like at this point, I'd set it up where the hatchling room is now, or what used to be the quarantine room is now like that super insulated hatchling room. And so I actually don't have stuff coming into the colony right now. But I used to have it where the first week would be closed off in a spare bathroom in the most sterile conditions. That's when I would do my first round of disease testing. And once it made it past that, it could then enter the quarantine room. The quarantine room, I would trickle stuff into quarantine over the span of like maybe 2 or 3 months. So everything would make it through that initial screening to make it into quarantine. And then I kept it in quarantine for like a year.
Wow.
So in that quarantine, I kept it like I would anything else in the regular colony. And, what I want to know is if that animal is going to exhibit signs of disease in the conditions I'm going to give it. And if it doesn't for a whole year, I feel pretty safe moving it into the colony, into, whatever other colony it's going to be. Now, again, I layer on top of that disease testing. So it has to make it, a first negative test to make it into quarantine in the first place. And then I'll test it a couple times, depending on where it's going in quarantine. But like, if you're just going to throw time at the problem, which I think is honestly not a bad way to do it, extending the length of your quarantine is like free. Like there's not, there's no cost associated with that. So I really don't see a downside of running out as long a quarantine as possible. But if you are doing anything less than 3 months, you're, riding a motorcycle with no helmet and like at 90 miles zooming between it's not like 90 days, which for many people is like a long quarantine. That is almost a recklessly short quarantine.
That's wild.
If you're going to do 6 months, I think at that point you're probably pretty safe. But like if you're doing a year, your chances of getting a surprise past that point are pretty low.
Okay. And go with babies if you can.
And go with babies if you can. Yeah, absolutely. Yeah. And, I like, it's a complicated world we live in. Like I would never be able to sell a snake out of that virus neutral building because I cannot prove to myself it's negative. I feel much more comfortable selling neonate animals. I feel much more comfortable buying neonate animals. Almost universally what we see in both the wild Burmese pythons and in captive pythons is that older adult pythons are where the highest viral prevalence exists. And that just makes sense. If you're a baby who's only been alive for a month, you're probably going to have a low exposure time. And then the other thing too that we see is exposure based upon the time of year. So we see that again in captive and the Burmese pythons, where in the summer, the viral prevalence was very low. In the winter, in the breeding season, when it's cold and stuff stressed, the viral prevalence goes much higher. And we see that in our captive pythons too. And so like, not only is that an animal that is young, so it has very low exposure time, but it's also hatching out at the time of the year when we see the lowest viral prevalence. And so like, baby animals are just a much different risk profile than an adult animal.
Did you? So your Burmese python that is in the biosecure building.
Yeah,
that is from the Everglades Park.
Yes. So the two of them that are in the building are, so I've two in the exhibition building that are ones that I personally removed from the Everglades. Mm-hmm. In the biosecure building, I have two other Burmese pythons, one of which is an animal I produced from my pair and kept back.
Oh, okay.
And then the other one is one that, that one was from a different set of experiments from actually a totally different organization, but they were doing pheromone studies and had collected a bunch of male Burmese pythons from Everglades National Park to use in these studies. And then one of them ended up laying a clutch of eggs. And so they were like, they asked me and they're like, can you like help us pull the eggs or incubate them or do something with them, especially because at the time they were concerned about virus in their colony. So it was kind of a good opportunity to create a sterile batch of baby Burmese pythons for them to work with.
That is so cool.
Right. Like I've been incredibly lucky to bump myself into cool scenarios. But yeah, so yeah, that is like the only legal Everglades National Park Burmese python from one of those eggs that I hatched out, helping them out as part of their stuff.
That's my favorite python, man, are the Burms. I really love them. I wish I could get one day. Maybe FWC will change the rules. I'm not going to hold my breath, but
yeah. Yeah, they've been, I remember the Burmese pythons were my first big snake when I first got the permit at the time to be able to keep big snakes under the previous laws, and it was just felt surreal to be able to walk into a room and legally see a Burmese python. Like it was, they are, especially one that came out of the Everglades. Like it just, they're fun snakes, and they're snakes that like. One of the things that I have had to like humbly accept is how little we understand about the animals in general, just from keeping them in a captive setting. Like I kept Burmese pythons for 5 or 6 years in a box, and I thought I understood Burmese pythons. And then I went down in the wild and did some of this, viral testing as part of this project, and talked with some of the other researchers, and the complexity of behaviors that they're able to document and describe to me are just mind blowing. Like they have patterns of these male, Burmese pythons hanging out in a summer feeding ground and then like moving towards like a winter breeding ground, and like really intense telemetry data that's basically painting really complex behaviors that we have no chance of ever appreciating when we keep them in a normal snake cage. So like, I always pause and try to remind myself that like the understanding we think we get from these animals comes from the context in which we keep them. And you might find a point in life when you get exposed to knowledge of that same animal outside of it in a different context, and the way you look at them is entirely different. So yeah, Burmese pythons are complex animals that are also just really fun captives. Like I love my Burms. They're really sweet, giant ball pythons for the most part, except for all the ways in which they are not.
Yeah, they're just so cool, and they're these titans they get so big, and what I really liked was the way your demeanor changed handling your Burm compared to the reticulated python.
Oh yeah,
which bit at my youngest son's face through the glass.
yeah. They are very different snakes. I have one Burmese python I've not been able to win over, but like they are an entirely different pace than especially some of the F1 retics and stuff that I have. But I do keep my stuff slimmer than most people, and I think a lot of the people who like, it's really easy to have a fat, tame retic when you treat them more appropriately with a more appropriate diet. They become a much more intense snake that is able to climb you straight like a telephone pole, and just like they are. I have a true appreciation for what an animal, a reticulated python, is. Like they are next level. The Burmese pythons seem to be a little bit easier to come to an understanding with for the most part.
Yeah.
Like they are, I feel, one more step towards domestication in some regards, at least in terms of like being very willing to be a placid captive, even when you are working with straight out of the wild animals. The retics take more of an understanding to make sure you are in line with that animal.
Yeah. Do you think that reticulated pythons should be bred at the level that they're bred at?
Oh, absolutely not. No. Yeah, reticulated pythons are honestly the biggest liability towards modern herpetoculture. Like straight up. Yeah. I mean, I love them. They're amazing species to work with. They are the ultimate python, but they're also incredibly fecund, and just dangerous. They really are. They really can be. Like, even ones that are, and there are very many very habituated retakes out there. So like I caution, like, I don't want to mischaracterize reticulated pythons, but at the same time, like, it is also very easy to mischaracterize reticulated pythons into forgetting that they are a true apex predator and should be treated always with that in the back of your mind. Like, they have killed people, they have killed and eaten people in the wild. Like, yes, you can get them to a point where they're very, very workable, but it makes me very nervous, the amount of trust people have in animals that should probably have a level of respect or danger cast over them more than they should. Like, they should never be a cheap, available snake. And it is very concerning that one successful propagation can represent, like, 50 animals. Like, it just, there's some animals that have a reproductive profile that works well in a captive setting to make sure that, like, market isn't oversaturated, or at least if it does, like with the ball pythons, it takes 20 years to get it to that point. Like, there's a lot of the incentives that are built into the industry that just don't seem to mesh well with reticulated pythons, that I think put them in a unique position of becoming a big liability for the reptile industry as a whole.
Does it terrify you when you see, like, the YouTube videos of these giant retics with, like, a child?
Yep,
with, like, a
absolutely.
very small child? It makes me feel so uneasy when I see that.
Yeah, yep, absolutely. Yeah, the visual that, like, was always left with me about that is not necessarily the true danger of it, because, in those circumstances, the people will swear by the animals and they probably are pretty safe. But all it takes is one small incident for it to be horrific. And with retics, there's just, it's easy for that to happen. But even if it doesn't, from a public perception image, like, most people will view a large python wrapped around a toddler's neck as no different than a toddler playing with, like, a gun. Like, it is a bad visual to leave the general public with if you're trying to live them, to win them over into thinking that, like, this is an animal that is kept responsibly in a captive setting.
Yeah. Yeah, it makes it hard to say that we're being responsible keepers.
Yeah. I mean, like, for me, the biggest, like, there's no other snake that we keep that reacts the same way male reticulated python does in the middle of breeding season if you smell like another retic. Like, Morelia do the whole, like, ritualized combat. Retics do the bite and shred. Like, they want
really
to, yeah, they become very aggressive and very hormonal in the breeding season. And, like, the type of bite that those male pythons aim to give are the type that require stitches. Like, it's not a puncture. It is almost like a bite and shred. Like, they are capable of hurting you and seem to be hormonally inclined during certain parts of the year, where, like, they really take contact from the keeper as another form of male aggression. And so if you smell like another male retic, all of a sudden your most tame animal can turn pretty dangerous. And like I said, it's not just the bite, it's the way they go about those bites. Like, the difference between a defensive bite and a feeding bite.
Mm-hmm.
There's a third category of bite that is occupied by male reticulated pythons.
I didn't even know that.
Yeah.
I did not.
That's
know
how
that.
I would describe it. Yeah.
I mean, thinking of the danger, I was thinking about just how my snakes get keyed up from when I pull rats out of the freezer in the garage and start thawing them in the garage. And then I come in to do something in the room. They already know something is up. You know, like they're all getting keyed up already. You know, my Timors are looking at me like, all right, dude, you know, like it's go time. So yeah, just the thought of you know, having a very small child with an enormous constrictor, like what if just somehow some prey smell comes that way?
Yeah, I mean the flip of the switch of these guys is potentially high, and that's true for almost all of these things. Like, as I have expanded my keeping and keep stuff in a variety of different habitats depending on the context of the animals, like, my appreciation for the complexity and depth of their behaviors has definitely grown. But it is probably, you know, it is dangerous to project too much emotion onto the animals, you know? Like, I really appreciate seeing a bunch of complex behaviors and clearly thinking that is going on in these animals, and clearly some sort of interest in me as a keeper and interacting with me as a keeper that they actually go and seek out. But like, it's very easy to take that feeling, which is real and true, and taking it a step further of saying like, oh, my snake loves me and wouldn't hurt me and understands that, you know, like there's
That's not a real thing.
Yeah, at least I cannot convince myself that it is. But, you know, by the same token, like, there are, like, my giant tortoises are a great example of, like, they seek out human attention. They like being scratched. They like people.
I saw this firsthand. Yeah.
But like, that doesn't feel like it has the same emotional impression in the same way that, like, it feels like my cat cares about me, you know?
Mm-hmm.
But all this stuff is on a spectrum of, and I've really tried to open myself up of what I think the spectrum of cognition is for these animals. And you really can learn a lot by even keeping the same species in different types of environments and seeing how they respond differently. And, you know, understanding that maybe it isn't the case that one is necessarily 100% better than the other in some regards, but that if you're looking to get the most out of your animal, that takes feedback into providing them an environment to bring that out of them, you know?
Mm-hmm. So you're referring to keeping in a larger, more elaborate, more naturalistic enclosure versus a tub?
Potentially, but you don't even have to take it that far. And so, like, one of the things that I did was I called it bioreactive, where I swapped out all the
TM.
tubs. Yeah, right. I swapped out the tubs for you know, clear tubs on an ARS rack, and stuck grow lights to the side of the cage, and grew out ball pythons in a normal CB70 rack, but with pothos and cork bark and isopods and a whole naturalistic setup, just shrunken down, you know, just in a tub instead of, you know, a big, you know, it wasn't the same thing as a big display cage, but inside of this tub, instead of having one empty tub with a water dish, it is, you know, there's a humid side and a not humid side, and the back is where the plant grows and the front is where the cork bark is. And like, I noticed really elaborate behavior just from giving them that diversity of stimulation in our existing setups. Like, you know, when they're in shed, they'll be at the humid part of the cage. Like, they're smart enough to exhibit a change in behavior based on their requirements if you give them an ability to change that behavior. And so Yeah, one of the things that I really enjoyed is putting cork bark in the blood python cages, because they
Mm.
loved moving it around and hiding under it and like hiding their head under it. And like, so like even just providing them a little bit of something, they made use out of that. And
Mm.
so, I try to make room for a spectrum of thinking, or at least not absolutist thinking of like, you know, tub is bad, natural is good. It's really just, you can provide them a spectrum of experience and you will bring out more complex behaviors in doing that, that probably almost certainly enriches that animal's life. But I also have data from really big python colonies that give him nothing but bare setups. And if you asked me to like find a problem with it, I couldn't find it in the data.
Yeah. But then you have the data too of the wild animals and their range and the
Exactly.
complexity of that behavior,
Yeah.
right?
Yeah. Yeah. So one of the things that I have tried to incorporate into how I'm thinking about it, at least, is like, we see differences in neurology of snakes that are given really stimulating setups and those that are given bare setups. Like, there's literally more thinking going on in those snakes in the diverse setup. I think that's a good thing, but that's just me applying an emotion to it. Like, what we're seeing is an extension of natural history. Like, many pythons have evolved to be stationary for 6 months at a time and not move because they're an ambush hunter. And so in that 6 months, what we know is that organ systems grow and shrink with snakes depending on, like, if they eat a big meal, their stomach will like sometimes literally double in size,
Mm.
um, among other organ systems. And so I think we're just seeing the same thing occur in the neurology. Is like, the same thing that makes a snake perfectly happy to sit in one spot for 6 months while it's waiting on an ambush trail is probably the same behavior that makes the snakes relatively content to live in a bare tub. But
Mm.
that's not the full suite of behavior that exists in that wild snake. Like, there's probably going to be times of the year when it's doing a lot more active hunting, a lot more moving around, and it's probably doing a lot more thinking in that process. So like, we know there are moments of really intense activity that occur in the digestive system when these animals are digesting food, and that shrinks when they are not needing it. And I sort of think the same thing is probably going on in these snakes, where they literally just aren't thinking about much when they don't have a reason to think about much. But if you give
Hmm.
them stuff to think about, then they have plenty to think about and they think about it. So,
Mm.
it's, again, I probably,
Do you think that we're
yeah.
taking advantage of that stationary mode that they have
Oh,
and
absolutely. Yeah, absolutely.
capitalizing on it and then depriving them of some facet of their life that they would have otherwise?
Yeah. I mean, I view it as no different than we are also selecting for animals that will eat European lab strain rodents,
Yeah.
like, there's, we are selecting animals to thrive in the context that we place them in. And so if you place them in the context of a bare box, the ones that survive and breed are going to be the ones that aren't really bothered by that. And so, yeah, we are, and that probably is coming from an extension of a natural behavior. And so we are absolutely taking advantage of that facet of their biology that allows us to put them in this type of setup and not see obvious consequences.
Mm.
But again, like, you see, if you give it to them, they'll use it. And so, it's, it's hard to not put emotion on, like, there's an exchange that occurs between how I feel seeing an animal act a certain way and what I think that animal is feeling from getting the opportunity to act in that way. Like, I don't know. I can say that I feel better when I see that range of behavior, but I can't say that, that represents a true, that is genuinely better. Like, it's not, if there's an aspect of their biology that allows them to be content to sit in the wild for 6 months at a time, I shouldn't not feel good about that behavior in much the same way. Like, I should feel just as good about that part of their natural biology as I should for the part of their natural biology that allows them to enjoy a enriched habitat. I guess.
Yeah, I mean, I like seeing them do snake stuff.
I like seeing them do snake stuff. Yeah, but I can appreciate that part of doing snake stuff in some circumstances is them sitting there without a requirement of stimulation.
Yeah.
And I don't think I can't say that a snake sitting on a log for 6 months is deprived of anything.
Yeah.
And so it's hard for me to say that a snake sitting in a tub is necessarily deprived of anything if it's kind of an extension of this part of their biology. But the joy I get is more so when I see snakes doing snake stuff. So it's like, that's why I'm like, I am projecting my own emotions onto the circumstance, but I am doing this because I am a keeper. And so that is part of the dynamic. Like,
yeah,
there's a relationship between the keeper and the kept. And I think what I try to explore is the range that dynamic can take.
What if you looked at it from the perspective that you're depriving them of the choice to sit on the log or go do snake stuff?
Yeah, I think that's entirely accurate. Yeah.
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 Yeah, because because that's like, if you find a snake that's been sitting under a log out in nature, well, he's chose to do that. Maybe he had a giant meal and, now he's solid. He doesn't want to go do snake stuff. But then you might see a black racer just like, slinking through a bush and falling out and, going all over the place and, and then he's doing snake stuff.
Exactly. Yeah. And that is probably, another thing that would that has helped me fall into this line of thinking, in the sense of like, you couldn't keep a black racer in a tub in the same way that you could a ball python. Like, they don't have a part of their biology that says, I'm happy to sit here for 6 months.
Yeah.
And so guess what is not commercially bred at scale in CB70 tubs all over the world, So it's like, the animals that do well in the context we give them, because they have natural behaviors that allow them to exist in the context we give them, are the ones that are successful. And so it is absolutely valid to say that depriving them, not giving them that choice is depriving them of that. And you also have to make space for the reality that you can deprive them of that and not see the consequence of it,
Yeah. It's a very complex kind of, because I keep my animals that are in quarantine in tubs and they do just fine in a ridiculously small amount of space. I had a couple of picky eaters though that once I put them into larger enclosures, they started eating rodents readily after only being on birds or being picky. But maybe that was just the change in stimulus. Maybe it was just something they needed to make them hungry. Maybe just moving around increase their metabolism. Who knows?
Yeah. I encourage people to hold whatever views they have pretty loosely and leave room for appreciation for the animals to grow and changes, understanding changes. And it's been very interesting watching the industry as a whole progress, in the sense of like, when I started in 2009, like I would say from 2009 to 2020 was really a first wave of reptile keeping. Not even a first wave, because that's probably the first wave of like industrialized reptile keeping, where it was a true industry scale, where we had, trade shows that occurred very regularly and stuff like that. And then in 2020, it really seemed like there was a big shift towards naturalistic keeping. And it has been really interesting watching the industry kind of segment itself from there. But this new second wave of keepers really do seem to invest in providing the best for their animals and really focusing in on the enjoyment they get from the aspect of keeping versus the enjoyment they get from the the commercial breeding or something like that. And so I think we will, and are currently experiencing, as seen in the prices of everything right now, like a shift of people really valuing investing in the animals they do have, instead of chasing whatever the next morph trend, whatever is. Like, it definitely seems like the new wave of keepers are really invested not just in the animal themselves, but in the context in which that animal exists. Whereas like before, it was a lot of times like just adding a new gene to a roster to fit into your mix and match set of mutant alleles.
Yeah.
But, it creates incentives that aren't necessarily in the animal's best interest. And now I feel like the animal's best interests are certainly a large part of the conversation.
Yeah. Yeah. And it's interesting even seeing like Dr. Julander kind of shift in that direction, where he wants to see more of his snakes doing more snake things. And he talks about it often, and he's changing the way he keeps and trying to get everything into cages. And so it is, I mean, and he's somebody who's been in the hobby for quite a while. So it's interesting seeing that change, I'm sure, from your perspective with all your time in the hobby, and then seeing it in keepers too that have so much experience in the hobby.
Yeah, absolutely. Yeah. I remember like we saw a group of wild spotted pythons in Australia.
Nice.
And one of the coolest things to me was finding a pair about 2 feet apart from each other, but not during breeding season. Like, they were clearly like localized to this specific hill because someone was like, this is where you go, you will find some spotted pythons here. And like that was their home. Their home was that side of that hill, and they lived right next to each other. And like, it really gave me an appreciation of the quasi-social nature of some of these animals and some of the complex behaviors that we're not really seeing in a captive setting, and how elaborate their behaviors can get when you provide them that. And then I got home and I set up my children's python in a big naturalistic setup, and it was just seeing it bask on the rock in a tank and seeing it bask on the rock in Australia. I was like, this is right.
And were you keeping them communally?
No, the cage was not big for that, but I do experiment with a fair bit of communal keeping for a lot of stuff, as you've seen.
Yeah.
But that obviously adds more complexity to it. But I have found that within reasonable bounds, these snakes are a little more social than we give them credit for. And leaning into that in a way that is responsible is a way to provide a lot of stimulation for multiple animals without like sacrificing on space. Like if you are, I'm able to provide a much bigger cage for two animals than I could half the size for each one of them, you know, as long as I'm able to meet an ability to separate them when I need to or whatever. So a lot of my cages are very fluid where they can be one cage or many cages. And so I can keep them totally separate some parts of the year or give them access to each other at other parts of the year. So they all have bigger cages. And so it's sort of I've really loosened my rigidity on strict ways of how to do things and given the animals a little bit more leeway in terms of what they are exposed to and kind of based upon the feedback of that. You know, I now no longer move animals to, you know, move the male to the females, at least in some of the buildings. I don't move the male to the female's cage. It's about removing the divider so they now occupy the same space. And I might even extend that into parts of the non-breeding season because that way they both have, you know, twice as much room, but are still having whatever needs met. As long as, you know, you can separate them for feeding and are aware that essentially you have to keep an eye out for problems occurring and have the ability to correct when problems are occurring. Like I can obviously split the cage in two. So it's, you know, I've definitely given myself a lot more freedom to experiment with caging and co-housing for some of this stuff within obviously reasonable bounds. There's so much of the stuff is nuanced that you worry about making too broad statements with it. But like, if you free yourself up from broad statements, you'll see that a lot of them are not necessarily built on anything that is like concrete. It's really just what has worked for other people. And you can build off that with insights from the animal to really get the most out of it.
You keep all your Halmahera scrubs together?
For the most part, for some parts of the year. Yeah. Yeah. So right now I have a group of 2.2 that live in two conjoined grow tents that, so I have the ability to separate them just from the two grow tents, but then I also have large cages, like large bins within those cages. So if I need to separate them out for short periods of time, I can. But then I also have backup cages. So if I really need to separate them, I can remove them entirely from the setup. But yeah, I've kept them communally now for 2 years without issues. I guess it's only been about 18 months. Yeah.
Do you think that that's what led to your success with breeding them?
It seems to. That seems to be because I think Chuck did a similar thing where he had individual cages for the animals, but connected through tubes that he was able to just allow the whole group to kind of commingle all together, at least during the breeding season. But yeah, I feel like I had good success using that. But I don't know that is the only way to do it by any stretch.
What do you, if you had to pick one thing, what do you think was the thing that got you to breed them successfully?
I mean, who knows? It's just luck. Like, I mean, a lot of it had to do with how I treated them in the first place. So like the first year they came in, I didn't touch them at all. Like they
wow.
So they went through that first quarantine step, but then their quarantine cage was a bioactive cage with a totally closed front that, the whole front was PVC, but I had a little eye hole that I could flip up so I could peek in there and look at them. And so like, aside from swapping out the water each week and like cleaning up the obvious, you know, turds. The rest of the time, I did not touch them at all for the first year. And at that point, they were really well established, feeding well. And then I started keeping them in individual cages, like you would a normal scrub python. They did really well for about 8 months or so in that setup. And then at that point, they were ready for an adult cage. And rather than build something up, I was like, oh, well, I think I'm just going to try one of these grow tents and set it up really well with, multi-tiered stuff like that. But yeah, I think a lot of the success was just hands off. And then the other thing too is like I really believe in seasonal feeding. So a lot of that came, like the ovulation that I got came immediately after a very heavy period of feeding. So seasonal feeding I view as a push-pull. So there's parts of the year where they're like not getting any food. They're fasting entirely, usually immediately before the breeding season. And then during breeding season, I will feed them twice as much, like almost to catch up for lost time. And so I'm giving them the signal of a season has changed and now you have a bunch of resources. And that really seems to be the trigger of breeding a lot of this stuff. Like you can almost food cycle anything at any time of year and get it to go. To some extent. I will obviously probably not the case for Hamaheras. They're very tricky to dial in, and I'm sure a lot of that was just luck. But I have found that seasonal feeding seems to be a very strong way to induce stuff to ovulate.
How heavy are you feeding them? Like, what does that look like?
I mean, so I think I don't really, a lot of this is just based off of my own attention. Like during the winter, I will probably not feed them so I don't think I fed them at all December, January. And then like maybe February or March is when I would start giving them back food. But like for the Hamaheras, I know the female that ovulated, I probably fed her 2 larges. Like I think I fed her a large a week for about 4 weeks. And then I thought what happened, well, I fed her a large rat for every week for like 4 or 5 weeks. And then she stopped eating. And I actually thought she was getting sick. Like not like respiratory sick, but like she just didn't look right. She went from eating to like not eating. The color was starting to darken. But I missed the ovulation. And eventually I decided I was going to take her out of the breeding setup because I was like, she's not eating. She's just stressed out. Set her up in an individual cage. And then when I was moving her into that cage, I decided to palpate her and then felt eggs inside of her and was like, oh, this is not her being sick. This is her going off of food and actually gravid. And so, yeah, she ended up laying 4 eggs and all 4 hatched and they're all doing great. Hopefully I'll have better success this year, or not better success, more success this year. I think one of my 2 females has a shot at going again, but I've honestly been so distracted that I've not had the chance to beef them up with as much food. So this will kind of be my experiment, is that I'm about a month late of where I was last year for them to go. So if I feed cycle them now, it'll be a proof of concept for me that a lot of their reproduction is tied more to food than strict seasonality.
Interesting. They're on my list. Those snakes are so cool. They're
a very fun species to work with in the sense of like, they don't act like other scrub pythons. They're very shy. Most of my adults now at this point are they're calmer than a lot of scrub pythons, but like the things about them are just a little goofy. Like they are more inclined to tuck their head, much like a ball python, than some of the other scrub pythons. Like my barnecks I can get away with stuff with the Hamahera pythons that I cannot get away with the barnecks.
Those babies were so cool. They haunt my dreams.
Yeah, that was a true dream to hatch out. And they hatched out in the middle of a hurricane. Like the hurricane had knocked out power. And
That's wild.
so I was actually nervous about the eggs getting cold. But yeah, they all have hurricane themed names because they hatched out in the middle of a power outage.
That's so cool. Yeah, I remember that, right? Typhoon and
Yeah.
yeah. How do they do the babies, do the offspring behave differently than the parents?
Not a whole lot. Like they were very shy. Like I remember the first set of neonate scrub pythons I got in were just like extremely wiry, uh, very bitey, and the Hamaharas instead would almost like again curl up their head like a ball python. Um, and even just the way they hold you is a little different. I don't really know how like a scrub python feels like it's holding on to you, whereas a Hamahara python feels like it's like draped on you.
Hmm.
Like I feel like the scrub pythons use you as a base to secure their body, and then their head is floating and paying attention. Whereas like the Hamahara pythons, I'll sometimes just have them kind of like nestle into my arm, I guess. More like a I don't know. Like I said, the way they hold you is different than other scrub pythons. And that extends from their shy nature. Like they just kind of have a shy vibe. They don't yeah, it's hard to describe. They're a weird python. They really are.
All right, man. We're almost on 3 hours just recording with this session here. I appreciate you being so generous with your time and all that. Oh, real quick, what about Timor pythons? Do you think you could keep those communally, or do the male Timors do the same thing that the other Malayo pythons do? Do they try to kill each other?
I actually don't know. I've not heard of people having anywhere near as many territorial type responses from Timor pythons, but there's also just so much fewer of them that it really doesn't. I don't know that there's a large enough sample set, but yeah, even those guys, like all of the big snakes would have challenges doing communally. It's definitely not impossible. What was the guy in I feel like the guy in England, was his name Dougie, has a bunch of
sure.
He has a really intense naturalistic setup for retics that I think might have multiple in that cage, but they are like room sized. Yeah. There's so I don't know about co-housing Timors, but if I were to go about doing it, I would have 2 large cages that had a removing wall and kind of go from there.
Yeah. Have you had success with your Timors?
I haven't tried. They're still pretty small.
Oh, okay.
They have the age, but I've just not fed them as heavily. So
okay.
They're probably ready.
My female is. Sorry, go ahead.
Oh yeah, I'm like been on the fence about them because they're like, I don't know, more. They're pretty different from the other stuff that I keep. So like, I'm trying to like sort it out where I have like a weird Python year and then like a commercial Python year. So I think next year I'm going to give a shot at some of the weird Pythons and make that my focus. So I might give them a shot then.
Cool. How old are they? I
think 4 or 5. Like I said, they have the age. I just don't feed anything near as heavily as other people. So all of my stuff is on every other week feeding for the most part.
Okay.
Yeah, I really, in order to feel the impact of extra food, you need to not feed as much as the hobby does as a standard, you know? Like if you're keeping them on a regular food schedule and you just escalate that, that is not the same thing as food cycling of where they are having a season that is not getting any food for quite a while, a season with ramped up food, and then a season that's just kind of regular feeding. So I try and keep things very dynamic, But I expect next year I'll have them. If I start with the focus of breeding them next year, I will be able to feed them in a way that will set them up for success.
My female is like, she's a 23 and she has like, I feel like she tripled in size, but realistically she probably doubled in size just since I kind of fasted her over the winter, gave her some time off, fed her more sparingly. And then, yeah, she's bigger than my 2022 male now.
Nice.
And she's just intimidating as hell because she always is expecting food now.
Interesting. Yeah, mine never, they seem more flighty than feedy. Like they know they'll give me a feed response if there's food in the room, but if it's just me as a keeper, they're all over the place.
Yeah, I get that too. I definitely get that too. But yeah, so it's when there's food in the room. If there's food in the room, it's an intense experience. Yeah, it's very intense. If you reproduce them, I want a female from you so I can have 2.2.
Absolutely, yeah. Yeah, I'm hoping I'll have luck with them. The pair that I have is captive bred, so like the more generations from the wild you get, the easier all this stuff is to work with.
Yeah, mine are all captive bred as well by Ryan Young.
Nice, that was cool.
All right, man. Well, thank you so much for coming on. I really appreciate it. You're a good dude. You're doing good work, and you're saving the hobby and our snakes. So yeah, you're incredibly valuable. Thank you, sir.
Absolutely. Thank you very much for having me. And I always like talking about the research and honestly being able to push good information out into the community. But I don't know. I always feel like I leave this with more people having more questions than answers. But that really is the truth of it. You know, we have more questions than answers.
Yeah. I mean, that's science, right?
Yep.
All right, man. Thank you.
Absolutely. Take care.